Good morning and greetings to all the delegates from all over the world. Let us start with our first talk for the day. And this is the day two of our 2 days international conference on advances in pharmaceutical medicinal chemistry 2022. So the first speaker for our day is Professor Alexander McCarroll. He is a Grolman Gillick Professor of Pharmaceutical Sciences from University of Maryland, USA. And he has completed his BS from University of Hawaii at Manoa, USA, and his BS is in chemistry. He has also done his Associate of Science in Biology from Rowan College of Gloucester County and PhD in Biochemistry from Rutgers University, USA. Further, he has taken training as a postdoctoral fellow in the Department of Medical Biophysics, Karolinska Institute at Sweden, and then another postdoc in Department of Chemistry at the Harvard University. His fields of specialization include computational chemistry, biophysics, empirical force field development, computer-aided drug design, to name a few. He has an extensive experience of more than 30 years of teaching, research, and managerial experience, and he has handled many significant responsibilities till date, including being the professor of pharmaceutical sciences at University of Maryland School of Pharmacy. So he had joined School of Pharmacy of University of Maryland in 1992. He's currently also the director of University of Maryland's Computer Aided Drug Design Center. He is also the co-founder and the CSO at Slix Bio LLC, a biotech company in the area of computer aided drug design and development. He has many publications to his credit. He is also the editorial board member of many international peer-reviewed journals. He has done many presentations and given many keynote addresses, including being a chairman of several conferences. He has published more than 750 peer-reviewed articles and has co-authored several books, book chapters. His H index is 107 and I-10 index of 393 with more than 83,000 citations to his published work. So we are very lucky to have him as our speaker. So I would like to welcome Professor Alex McCarroll. And due to some reasons, he was not available to give a live talk. So he has sent us a recording and we'll just be playing that recording for all the delegates. Thank you so much. Stay tuned. Before we start, let me also welcome our next speaker, Professor Jose Medina. He's from Mexico. And thank you so much, sir, for joining us.
Oh, thank you so much for the kind invitation. Thank you.
Okay. All right, so let us start with our first speaker, Professor Alex McCarroll. I'll be sharing his talk now. Can the IT department confirm if the talk is visible?
Yes, sir.
All right. Thank you so much. So I'll start the recording now. I'll start playing now. IT department, please ensure that the recording is on.
Hello, my name is Alex McCarroll. I'm a faculty member at the School of Pharmacy in the University of Maryland, Baltimore. I am in the Department of Pharmaceutical Sciences, and we also have a startup company called Silks Bio LLC. And today I'll be talking about the Silks technology. Associated with that, I have my conflict of interest slide to let you know that I am involved in a startup company as well as have site identification by ligand competitive saturation. We've also developed another technology. I'm not going to talk about single-step phase perturbation. And then I have one slide on the charm general force field and the charm general force field program. And this is important because if we're going to look at hundreds or thousands of ligands on a daily basis, we've got to generate the topologies and the parameters for those to allow us to do the molecular dynamic simulations in the CGNFF program. And just briefly, the CGNFF program allows us to input, say, a MOL2 file. It goes through the CGNFF program and then outputs the ligand topology information along with the parameters. And the CGNFF program allows this to happen instantaneously. It's so fast that we can just take any new molecule, generate the topology and the parameters, do whatever computations we want, and then delete all those files because we can just generate them rapidly again the next time. So this really facilitates the drug design process. This is an important tool, although not the focus of this talk. So now we get to the site identification by ligand competitive saturation technology. Now this is an approach which allows us to generate a functional group affinity pattern for the entire protein. This is the external regions of the protein as well as the internal regions of the protein. We do this using a combination of grand canonical Monte Carlo MD simulations. I'll introduce GCMC a little bit later. And importantly, when we do these simulations, we include protein flexibility. We have an explicit solvent representation, and we have an explicit solute representation, with those solutes having different types of functional groups that allows us to generate this functional group affinity pattern, which we refer to as the frag maps. These frag maps are essentially our pre-computed ensemble that we then use over and over again when we start to actually do the drug design. Now, basically, in the Silks methodology, what we're doing is we're taking some types of solutes, say benzene, methanol, we put them into an aqueous solution, and then we do our exhaustive simulations. And so the benzene, methanol, or a collection of benzene, methanol, other solutes will diffuse throughout the system. And again, this is in the presence of water. And then we can calculate the probability distribution of the solutes throughout the simulation system. If it's just the solutes in water, this will be a homogeneous distribution. But then we can add in a protein or other macromolecule, do our exhaustive simulations, now calculate a new probability distribution in the presence of the protein. And what we see is that we now have higher probabilities in regions in favorable binding pockets. We have lower probabilities on top of the protein where the solutes can't go, or in unfavorable pockets. And then away from the protein, we have a probability distribution, which is basically the same as that in solution. So what we next do is we take the probability distribution in the presence of the protein and normalize it by that in the absence. So we end up with a normalized probability distribution where the probability is a value of 1 away from the protein, which is equivalent to being in solution. It's greater than 1 in a favorable binding site and less than 1 in an unfavorable binding site. So now we have these probability distributions, which is telling us where different types of functional groups want to be located, whether they want for them to be favorable or unfavorable, and this is all relative to solution. So by doing this and having explicit water, we're essentially taking into account the energetic cost of desolvating the protein binding site. The normalization takes into account the energetic cost of desolvating the functional groups. We include protein flexibility in our simulation, and then obviously our probability distribution includes contributions from the favorable or unfavorable interactions between the different types of functional groups and the protein itself. So do the silk simulation in Some allosteric modulators. So here I show on the structure of this GPCR, the position of two different allosteric modulators. So one of the things that we want to do with our computational technique is identify novel binding sites, which we never even considered before. And the Silks technology is quite good for this. Another problem is we want to be able to understand very deep pockets, such as an orthosteric site for a GPCR, which is very deep. When we look in the side, we can see the transmembrane helices. The ligand for the protein is deep within the binding pocket. If we look down from the top, we can see that it's almost totally occluded. And so what we want to do is we want to be able to sample our solutes and get the functional group affinity pattern, the frag maps in these deep pockets, as well as on the surface of the protein. So the problem with doing this is that we have limitations associated with the rate of diffusion of the organic solutes in our system from the exterior of the protein to the interior and in again and out again multiple times, as we need to calculate an effective equilibrium between the bound and the unbound states. Also, when we have deep pockets like this, we need to have conformational changes in the protein that allow the protein binding site to become accessible. So that becomes another limiting effect. And a great example of this is like the androgen receptor, the nuclear receptors in general, where the binding pocket is totally on the interior of the protein. So the protein has to actually partially unfold for the ligands to move in and out of the binding pocket. So this obviously occurs on very long time scales, which is not directly accessible in molecular dynamics simulations. So what we've done to get over this problem is use the grand canonical ensemble. And in the grand canonical ensemble, it's a constant chemical potential, volume, and temperature ensemble. But what we're doing is we're actually inserting and removing particles, which in this case would be water or solutes, in and out of the simulation system using a Monte Carlo algorithm where the insertions, deletions, as well as rotations and translations of these solutes are accepted and rejected based on a metropolis criteria. Here's the equations. I'm not going to go into the details of that now. And this is powerful because we can now start to insert solutes directly into the system so they can be actually inserted into, say, the center of a protein in a very deep pocket and so on. And an example of the GCMC simulation of just simply benzene and water is if we take benzene, we assign it an excess chemical potential of 0.8 kilocalories per mole, which corresponds to its free energy, and we do that in an explicit water environment, we will end up with a final concentration of benzene in the simulation system of about 1 molar, which corresponds to the standard state that we're using. Now, one of the problems of GCMC is actually taking a solute of the size of benzene and put it into a box of water or a sphere of water. So to overcome this, we developed a methodology where we have what we call an oscillating chemical potential, okay? And what the oscillating chemical potential does is it actually turns the chemical potential of a specific solute up or down in order to get enough successful insertions into the simulation so that we can achieve our target concentration. So basically what this is basically it's enhanced sampling in chemical potential space, which allows us to study the presence of different types of solutes, benzene, propane, acetate, and so on, in a condensed phased explicit solvent environment, which we can now use to study complex biological molecules such as GPCRs and so on. So this is a very important part of the technology, which we then combine with the molecular dynamics simulations. So our overall SILCS grand canonical Monte Carlo MD sampling protocol is basically we go in, we put the protein, whatever system we're interested in, into aqueous solution with the collection of the 8 solutes at about 0.25 molar. We then go in, we do 200,000 Monte Carlo steps of our oscillating chemical potential GCMC to re-sample the distribution of the water and the solutes in the simulation system. We then stop. We do 1 nanoseconds of molecular dynamics simulation using the program GROMACS. We then stop. We now do another 200,000 GCMC steps, followed by another MD. We repeat this for 100 cycles. So for each individual silk simulation, we get 100 nanoseconds of MD sampling, from which we get our probability distributions of the water, the solutes, the protein conformations, and so on. So this is our pre-computed ensemble. We get adequate conversions. We basically do 10 individual silk simulations, from which we get a total of 1 microsecond of molecular dynamics. And then again, from this, we get our resulting frag maps, which is our pre-computed ensemble. And typically a calculation like this on a GPCR with explicit solvent, the lipid bilayer, takes about 3 days on 10 2080 Ti GPUs. So it's a big upfront calculation, but once we've done this, we can now do all of our ligand design very rapidly, as well as have nice qualitative information about the to help our computer-aided drug design efforts. So basically what we've done, here's an example with a phosphatase 1B, where now we can look at the distribution of the frag maps. Again, these are favorable frag maps in and around the entire protein. You can see the different classes of functional groups in different colors. And again, you see maps over the entire protein surface, as well as maps inside of the binding site of the protein, the active site. So these frag maps are what we now start to use for our ligand design and development. Now we can move into the binding site. You can see here we've got a ligand where we've now overlaid the ligand in its crystallographic orientation on top of the frag maps. We can see how different functional groups in the ligand correspond to the different frag maps. We see aromatic and aliphatic frag maps overlaying the aromatic and the aliphatic groups in the ligand. Down here, we see orange negatively charged maps, which correspond to the acid groups and so on. And now right away, you can start to see that how these different types of frag maps recapitulate the functional groups, but you can also start to see how the frag maps can give you information on where to add functional groups to the protein to better occupy the frag maps. So you can see here, I could add additional, say, aliphatic groups. I could add an acceptor group. I could add, say, an O-methyl group, take advantage of this acceptor region, methyl groups reason. So this is the qualitative information that we can use for ligand design. Now, what I'm showing here is the solvent accessible surface of the protein from the crystal structure. But because we have protein flexibility in the silk simulations, we actually move beyond that. And so what I'm showing here on the left is, again, the same image where we have the frag maps, the ligand, on the solvent accessible surface. If you look at this solvent accessible surface here in particular, you can see how the frag maps actually penetrate underneath the surface. And this is because it's now, these simulations show us how the protein can change its local conformation to allow for ligands to bind essentially underneath the protein surface. And we can define this in what we call the exclusion maps. And the exclusion maps are basically the region of the protein in which the water or the solutes never sample. It's like the totally forbidden region. And if you compare your solvent accessible surface with that of the exclusion maps, you can see how much the protein can open. And this is giving us an idea of how much the protein can open when we do our ligand design. So once we're doing our ligand design, we get rid of the crystal structure, because now we can readily see how we can sort of build the molecule out this way, out that way, and so on. And the protein, we know that the protein can relax to allow for this, the ligand, a bigger ligand to be built, and these types of things. Another important aspect of ligand design is knowing the position of water molecules. If we have water molecules in the binding site, we have to displace them. Are those water molecules easy to displace, or are they difficult to displace? What I'm showing here are water frag maps in red. And this is from a simulation with the protein 1FJS. I've then overlaid water positions from another protein structures for the same protein, but different PDB. And you can see how the silks results recapitulate the positions of the waters. But what's interesting now is we bring in the other frag maps for the different solutes. We can see positions where the different solutes can actually compete with the water molecules to kick them out. So when we're doing our ligand design, we would know that these water positions can actually, those waters can be displaced. Whereas down here, we see water molecules, which there are no frag maps for any other solutes. So these water molecules would be difficult to displace. Again, this is more qualitative information that we can use for our ligand design and so on. Now we can go from qualitative design to quantitative design. And simply what we're doing here now is we're taking our probability distribution and Boltzmann transforming that into a free energy. And we call these the grid free energies. So basically we now have energy maps on 1 angstrom cubic voxels, which surround the entire protein, encompassing protein interior and so on. So we can now think in terms of energetics. And here I'm showing these grid free energy scores for aromatic groups at a cutoff of say -1.5, where we're seeing the most favorable regions for an aromatic group, and then at a lower cutoff of say -0.5 or less favorable cutoff. And you see the spread maps are so spread out. So we can now start to look at these frag maps and think in terms of energetics. And what we can now do is we can now calculate the binding affinities of ligands. And so basically what you do is you take your ligand, you overlay it on the frag maps for each atom in the ligand. The experimental binding affinity. It's not directly the experimental binding affinity, as there's certain approximations and certain terms that are left out, but it's now the type of energy that we can use for making quantitative predictions about changes of affinity, about the absolute binding affinity, and so on. And what's nice here is that because we already have the frag maps, we've pre-computed those. The ligand grid free energy calculation is essentially instantaneous. So this allows us to get to large numbers of molecules and so on. And then once we've got these ligand grid free energies, we can now basically take a molecule. For the molecule, we can actually start to do conformational sampling of the molecule in the field of the frag maps, from which we can now calculate ligand grid free energies for say different molecules. We can start to do correlation analysis and these types of things. And so we can do this Monte Carlo calculation. This can be very local sampling of just let the molecule relax in the local region, or we can actually fully rotate the molecule to do like a very extensive type of docking type of Monte Carlo calculation and so on. And once we've got that, we can start to now correlate our ligand grid free energies with very experimental observables. This is a published study on the protein BCL6. We're looking at a number of thioureas, and you can start to get nice correlations. Once you have nice correlations, this is showing you how you can quantitatively use these frag maps and the ligand grid free energies to predict relative binding affinities and so on. And what's an additional tool is we can step back a little bit is like we can calculate the change in the binding affinity. Here we have two changes of a molecule where we're going from an O-methyl group to a sulfonamide group. We see a nice change in the binding, becomes more favorable by almost 1.5 kilocalories. And this silks corresponds to that quite well. So this is the overall LGFE change. But what's nice here is we can actually go in and we can actually look at the scores of the individual atoms, and then we can do a summation over the atoms in individual functional groups. And we can see that when we go from like an O-methyl group to a sulfonamide group, we actually go from a contribution of like -0.3 to -4. So this is a huge change. So this sulfonamide group really has very favorable interactions with the protein. However, this gain of energy is much more than the overall binding difference. And what we see as we look at other parts of the molecule is that while we gain binding affinity here, in the cyclohexyl group, we actually lose it. We go from -1.4 to -0.2. So while we've gained binding on one end of the molecule, we lose it on the other end. So we have this highly detailed quantitative information on the contributions of different regions of the molecule to the overall change in the free energy. And obviously this is very good information that we can use for our ligand design and development. So this is very powerful, and it shows just the great information content of the Silks technology, which is not available from like free energy perturbation or other computational methods and so on. Now, one of the issues is that when we have these frag maps, and I won't go into this in detail, but we can actually, once we have frag maps, we start to do a medicinal chemistry study, we start to have some experimental data, we can actually use a Bayesian Monte Carlo machine learning approach to actually optimize the frag maps to actually improve the predictability of the model. So we have this Bayesian Monte Carlo machine learning approach, which allows us to improve the predictability of the frag maps. Even in a low data environment, we can have a relatively small number of compounds where we can start to optimize the predictability of the Silks model. And again, you can go back to the original studies, but basically you can increase the predictability. We can see the original maps, and then we use this Bayesian machine learning approach to increase various metrics and so on. So it's a powerful approach. And again, it indicates that our frag maps are actually pretty predictive from the beginning. We can even improve on that predictability as we go through a medicinal chemistry study. Now, how does the Silks technology compare to free energy perturbation? Well, we undertook a study where we studied 8 different targets, which had been subjected to previous studies, which were published in JACS. And in the original study published in JACS, they looked at a number of ligands for each of the 8 targets, a total of about 199 ligands. So we basically went back and applied our technology to this set of molecules to compare with directly with free energy perturbation. And then what we also did is actually, in the study, there's actually over 400 ligands that were available. We actually then went on to the full 400. And this paper has been published in Chemical Science 2021. And basically, we can start to compare the Silks methodology, where we have the mean unsighted error between the experimental and the calculated results, with the correlation coefficient and then the percent correct. This is true positives and true negatives. We can see that the Silks default method is quite good with respect to other free energy perturbation methods. It's quite comparable with the other free energy perturbation methods. And then when we start to do our optimization approach, in particular our Silks machine learning approach, we actually get the predictability of the Silks methodology is actually improved over the free energy perturbation techniques. And the silk simulation results, once we have the silk simulations done, these calculations are hundreds of times, if not 1,000 times faster than the free energy perturbation methods. So basically, we've got a computational method which gives us quantitative accuracy of free energy perturbation, but it's hundreds to 1,000 times faster than free energy perturbation. And we can see just the various correlation plots up here at the top. This is so we can now start to use this in a drug design context with the reliability that we're getting predictions which are as accurate as free energy perturbation. And notably, when we extended our method to the full 400 ligands, we still maintain our predictability. And the free energy perturbations were never done on all 400 ligands, probably to a large extent because the functional group additions were quite large and so on. With the silks technology, we can add very large functional groups. It's quite flexible. Now, for the rest of the talk, a little bit more, I just want to go over some studies we've done with the beta adrenergic receptor, it's a GPCR. Here's the silk simulation setup. Stay in time. I'm going to sort of move ahead pretty quickly. What was nice with this system is we had conformational, crystallographic conformations for both the active and inactive structure of the protein. So what we did is we went in, we did the silk simulations on both conformations, the inactive form and the active form of the protein. We had two sets of frag maps. We're now looking into the orthosteric site of the protein. And once we've got these frag maps for the two different conformations, we can actually calculate difference frag maps. So what the difference frag maps are actually showing us where different functional groups should be to facilitate agonism versus antagonism and vice versa. So this is interesting because you can think of the frag maps as being of utility to calculate binding affinities, but now by focusing in on the given conformations, we can also use them to be predictive of agonists versus antagonists, and this is very powerful. And here I'm just showing that when we take the two types of maps, we have the active frag maps versus the inactive frag maps. The active frag maps, when we look at the predictability with agonists or partial agonists, we see nice predictability. We looked at the inactive maps with the agonists and partial agonists, we see lower predictability. But now when we take the inactive maps and we look at antagonists and inverse agonists, we see better predictability. Whereas with those same molecules with the active maps, we see lower predictability. So basically we're seeing that we're getting these quantitative results with respect to agonism versus antagonism by taking advantage of frag maps on the active form of the protein versus the inactive form. So now what we want to do is we want to go in and we actually want to identify new agonists using database screening. So what we can do with the silks methodology is we can convert our fragment maps to pharmacophore features. And now these pharmacophore features can now readily be used to do database screening. And so what we now did is we took these pharmacophore features, we went in, we screened a database of about 2 million compounds. Based on the pharmacophore screen, we selected the top 50,000 compounds based on the structural overlap of the pharmacophore with the ligands in the database. We then took the top 50,000 compounds, we docked them against both the active and the inactive conformations of the beta adrenergic receptor. We did an initial round of screening using AutoDoc Vena, where based on delta energies versus the active and inactive form, we selected the top 1,000 compounds. We then did our silks Monte Carlo calculations, looking at the change in the free energy, the ligand grid free energy of the inactive versus the inactive form, from which we selected about 100 compounds. Analysis of those 100 compounds for like bioavailability considerations as well as chemical diversity led to selection of 15 compounds. And when we looked at those 15 compounds experimentally with our collaborators, we saw that 7 of the 15 compounds were indeed agonists. This is compared to isoproterenol, which is a known, it's a therapeutic agent actually for the treatment of asthma. based on being a beta-2 adrenergic activator. So this is showing just sort of the power of the technique where we can actually predict agonists as well as for the GPCR. Now, I'm getting towards the end of the talk. So briefly, I'm just going to sort of show some slides from some papers where we have what's called the Silks hotspot approach. This is a method which allows us to go and identify novel binding sites on a protein, which can be allosteric sites. We've now applied that technology to a number of systems, one of which is beta-glucosidase, where we identified allosteric binding sites for glucose 6-phosphate. We identified several sites which were then ultimately verified using mutation studies and so on. Another study was on the protein heme oxygenase, where heme oxygenase is involved in the breakdown of heme by bacteria. And it was interesting in this study, they were actually designing ligands to bind to the heme binding site of the protein. And we went in and we did our Silk simulations. We had the maps for the entire protein surface. And what we saw was that actually the ligands had a favorable binding site on the back of the protein, as well as in the heme site. And when we looked at the ligand grid free energy scores on this site on the back of the protein, we saw that they were systematically more favorable than the red scores in the heme binding site. And indeed, when these ligands were binding to this site, they actually caused inhibition, so they were functioning allosterically. We were able to verify that the ligands were binding to this allosteric site using hydrogen deuterium exchange mass spectrometry. So this is another nice example of where we can identify a novel site using Silks. A couple other technologies, we've now extended it to calculate permeation across lipid bilayers. We're using it to calculate the ability of compounds to bind to the H-erg protein. This is a drug liability issue. We're also using it to calculate protein-protein interactions. And then finally, we can now use this in combination to start to do formulation development of biologics, including antibody excipient interactions and so on. And then finally, just to acknowledge all the people in the lab who've worked on this over the years. It's been a lot of work by a lot of people. It's worked out quite well. Here's our experimental collaborators on beta-2 adrenergic, heme oxygenase, and then financial support. And again, the technology is available to industry through Silks Bio LLC. Thank you.
Thank you so much, Professor Alex, for that wonderful talk. And we are very proud to have you here live. So for the delegates.
I'm awake.
Thank you. So for the delegates, we had just played the recording, but then Professor Alex is here live now. So we'll quickly move on to the question answer session. And let me request my IT team to let me know if there are questions in the YouTube chat box.
No, sir, no questions.
All right. All right. Thank you
Come
so
on,
much.
somebody's got to have a question. I just got up especially for this.
Thank you so much, sir. Thank you so much. That was actually a wonderful, informative, and a stimulating talk. Really, it was wonderful to hear from you. And the take-home message from your talk would be, MD simulations generated frag maps and the related technologies which are based on Silks and GCMC can help in identification of binding sites, which may be superficial or maybe deep, located deep in the binding pockets, as well as these technologies can be used for qualitative and quantitative ligand design. So thank you so much, Professor Alex, for this wonderful talk and the stimulating talk, I should say. I just request the IT team to, So before you joined in, sir, we had displayed your bio, and I had chalked out the important points in your bio, and I had explained it to the delegates. So Thank you so much. And as a token of appreciation, we would like to present you virtual bouquets of pink roses and orange roses. So the orange roses, they symbolize the enthusiasm and the passion that we see in you and your work. And the pink roses, that they symbolize our gratitude and admiration for you and your work. Thank you so much, sir. And the e-certificate of appreciation shall be delivered to your inbox very, very soon. And thank you for your time and efforts, for recording the talk, sending it to us, and then getting up early and attending the conference live. Thank you so much.
Very good. It's my pleasure to put the talk together, and I hope the rest of the meeting is stimulating and everyone stays wide awake, because I'm going to go back to bed. Take care.
Thank you. Take care. Bye.
Bye-bye.
IT team, our next speaker is here, Professor Medina Franco, and he's from Mexico. So I'll request you to display the bio. Yeah, thank you. So Professor Jose, can we start?
Fine. Yes. So I'm gonna start sharing the.
Yeah. So just give me a moment.
Sure.
Yeah. So let me introduce Professor Jose to the entire gathering. So just a moment, sir. If you can just unshare your PPT. Yeah, IT team. Thank you. So let me introduce Professor Jose to the entire gathering. Professor Jose has completed his BSc in chemistry from National Autonomous University of Mexico, his MSc in chemistry as well as PhD in chemistry from National Autonomous University of Mexico. Further, he has taken training as a postdoc in University of Arizona. He has been an assistant member of the Torrey Pines Institute of Molecular Studies. He has carried out research work in some of the best laboratories in the world in Madrid, Spain, University of North Carolina at Chapel Hill, and University of Arizona, USA. He has been named a Fellow of the Royal Society of Chemistry, UK in 2017. His fields of specialization include his expertise in computer-aided drug design, molecular modeling, chemoinformatics, to name a few. He has held many significant responsibilities in the past. Currently, he is working as a professor and he is heading the Diaphragm Research Group at National Autonomous University of Mexico. So that is briefed here as UNAM. He is also a research collaborator of the Mayo Clinic, and he has been principal investigator and full-time research professor at UNAM. And he has many presentations and publications to his credit. He has delivered 40 invited talks in 5 different countries. He has published more than 60 abstracts in scientific meetings. He is associated editor for many peer-reviewed journals, including RSC Advances. He is a member of editorial board of 3 scientific journals, including the Journal of Current Pharmaceutical Design. He has edited and co-edited the books Epi-Informatics and Food Informatics. He has been a PI in several research grants, which are funded by government institutions as well as by pharmaceutical companies. He has supervised the research of 8 postdoctoral fellows, 11 graduate students, and 14 undergraduate students. And we are indeed blessed to have you here, sir. So Professor Jose would be presenting his talk on modeling and computer-guided discovery of inhibitors of DNA methyltransferase. Okay, just before I end, the last line that was not visible on the screen, he has published more than 185 peer-reviewed papers and 19 book chapters, 1 international patent, and his current H-index is 47. His I-10 index is 187, and he has more than 8,900 citations to his published work. So we welcome you, Professor Jose, for your talk on modeling and computer-guided discovery of inhibitors of DNA methyltransferase. Welcome, sir. The platform is all yours.
Thank you so much. Thank you so much for the kind introduction. Many thanks, Dr. Harris, for and all the committee for the nice invitation to participate on this conference. Good morning in India to everybody. And today I'm going to talk about, as Dr. Harris mentioned, the work of our research group focused on modeling and computer-guided discovery of inhibitors of DNA methyltransferases and other epigenetic targets. This is the outline of the talk. First, I'm going to present a brief background of epigenetics and inhibitors of DNA methyltransferases, or DNMTs. Then I will mention the long-term goal and the specific aims and strategies of this project. Then I will move to the results part, focused on the screening of compound databases and the prediction of epigenetic activity of small molecules. At the end, I'm going to present a brief summary and perspectives. First of all, I'd like to introduce briefly the research group, the research team at UNAM in Mexico. And as I will go to the presentation, I will be mentioning the specific contributions that the group has been making in this project. So in advance, many thanks to the research group that are actually presenting on their behalf. So very briefly, as a kind of introduction, epigenetics are modifications that happen to the chromosomes without alteration of the DNA sequence that lead to a stable phenotype. There are three major epigenetic target families: histone acetyltransferases, histone deacetylases, and bromodomains that are representatives of epigenetic writers, erasers, and readers. We know that histone methylation and acetylations are essential to the normal function of cells, and dysregulation on this process are associated with cancer and other diseases. So over the past few years, we have noticed, and this is a literature review, an increase in the number of publications linking epigenetics with different diseases, like cancer, that is currently the most studied, but also diabetes, neurodegenerative diseases, cardiovascular diseases, and others. Also, in the past few years, in the bottom graph, we see an increase in the efforts of the scientific community using epigenetics to identify potential drug candidates. And very recently, the past 5 years, more or less, also we've seen an increase in the number of computational applications to these epigenetic drug discovery efforts. Currently, there are 8 small molecules targeting epigenetic targets approved for clinical applications. These are called epidrugs. And there are several others, about 30 small molecules that are on the clinical development. On the bottom part of the slide are shown the chemical structures of some of them, and a comprehensive review of the current status of epigenetic drug discovery is at the bottom of this slide. As I said, one of these epigenetic modifications is DNA methylation. DNA methylation is basically the addition of a methyl group to the carbon number 5 of cytosine residues. This process is mediated by a family of enzymes called DNA methyltransferases. In cancer cells, we see an overexpression of DNA methyltransferases. Specifically, it is observed hypermethylation of tumor suppressor genes. There are a couple of de novo methyltransferases, DNMT3A and DNMT3B, and one maintenance methyltransferases, that is DNMT1. All of these methyltransferases have a regulatory domain, and most of them have also a catalytic domain, except the NMT3L. Over the past, like 15 years, several research groups have been crystallizing DNA methyltransferases. In 2011 was published the first catalytic domain, or the crystal structure of the catalytic domain of a human methyltransferase. And very recently, last year, was published the first crystallographic structure of DNA methyltransferases bound to a small molecule that is actually a quite selective inhibitor, the NMT1. On this slide is shown the chemical structure of this inhibitor. This publication was by GlaxoSmithKline. This slide are shown the chemical structures of representative inhibitors of DNA methyltransferases and other small molecules that are associated with the methylating activity. Especially, I would like to emphasize a couple of inhibitors shown here at the top left part of the slide are cytosine analogs that are actually approved by the FDA for clinical applications. Are quite potent inhibitors, but because of the nature of the chemical structure, are also quite cytotoxic. And therefore, there is a huge interest to identify other small molecules that are also potent inhibitors, selective inhibitors, but they have different chemical scaffolds. So here on the slide, in green are represented a couple of natural products. There are several others, drugs approved for other indications, the small molecules identified from computational approaches, here in red. We will come back to these compounds later. Also, there are several groups working on synthesis, medicinal chemistry. There are also several compounds with quite interesting activity, and additional molecules identified from high-throughput screening. Actually, the chemical structure from Glaxo that I showed in the previous slide, I'm showing again here, was identified using high-throughput screening of a corporate library of about 8 million molecules. So with this brief background, the goal of our research program is to identify and develop novel inhibitors with novel chemical scaffolds. The main strategy is using computational methods integrated with experimental validation. Specifically, one of the specific goals is first to understand the structure activity relationships of known active molecules. Other specific aim is to identify novel inhibitors or even modulators of DNMTs. A third one is the optimization of those compounds, and finally, drug repurposing. Today, I'm going to focus especially on the identification of novel inhibitors. And to achieve this specific aim, we're basically doing computational screening of compound databases. So the computational screening of compound databases can be organized in 4 major steps. First, it's the in silico characterization. We can call it like purification. Technically speaking, is data curation of the screening libraries. The second step is the actual computational screening or filtering of these compound databases. Third step is the experimental screening of selected compounds. And finally, the optimization of the lead molecules. Concerning the compound libraries that have been screened, we're pursuing different libraries like natural products, food chemicals, in-house libraries from collaborators, commercial synthetic libraries, and focused libraries. For the second step, the computational screening, also using different strategies like similarity searching, molecular docking and molecular dynamics, and machine learning. Currently, for the experimental screening, we are first using enzymatic inhibition assays to select the most promising compounds and additional experiments. And we're always open for collaborations. It would be great to collaborate with research groups in India. And for the optimization, also in collaboration, we're doing a chemical synthesis and testing of analogs. So more specifically concerning the compound libraries, these are three major natural products and food chemical libraries. One of them is the coconut. It's a free open library of natural products containing roughly 500,000 molecules. We're also screening public database natural products from Mexico that is called Biofakim, contains around 530 natural products, and food database. This is the database assembled by the group in Canada, also freely available, that contains around 22,000 food chemicals. Data sets of collaborators. Here are just a couple of examples. Synthetic analogs from collaborators in Mexico, Dr. Laura Alvarez and Mayra Antunes, and small molecules that are being synthesized by the group of Alexander Gagnon in Canada. Concerning the commercial libraries, the major source is Zinc database. And also, more recently, we have been exploring focused libraries. There's several chemical companies that are putting together, assembling collections of synthetic compounds that are pre-filtered and are enriched with potential compounds hitting epigenetic targets. And we will come back to these focused libraries in a moment. So the first filtering or computational screening that we did was actually a few years ago, doing the virtual screening of an open library, the National Cancer Institute compound collection, with more than 260,000 molecules. Here we implemented a sequential or cascade filtering approach using first filtering of compounds that we know have very few potential to be active compounds. Then the filtered molecules were subject to sequential docking approaches using different docking programs. And at the end, we selected 24 consensus hits. Of these 24 consensus hits, 15 were actually physically available for experimental testing. So out of these 15 tested compounds, 7 of them, here are shown the chemical structures, show inhibition of DNMT1, inhibition of the catalytic activity. So a couple of nice features of these chemical structures is that the chemical scaffolds were different from the chemical scaffolds of the known inhibitors published at the time. Also, most of these hit compounds have carboxylic acids. One of the hits from this initial screening was actually a natural product, nanomycin. This natural product turned out to be fairly potent with DNMT3B on an IC50 or 500 nanomolar, and actually was also selective for DNMT3B because this compound did not inhibit DNMT1. In addition, the natural product, nanomycin, also reduced the methylation levels of cytosine residues in cell-based assays in different cancer cells. Going back to the list of the actual compounds, the virtual screening active compounds, in the next few years were used as a starting point for the optimization programs. For example, this hit compound 14778 was later used as a starting point of a similarity searching of approved drugs. And from this project, we identify oxalacine, that, as you can see in the slide, is quite similar in the chemical structure to the parent molecule, and oxalacine also was a potent inhibitor of DNA methyltransferases. Independently, the group of Professor Gagnon in Canada took as a starting point the chemical structure of this hit, 745, and also his research group improved the potency of this molecule. Other research group, in this case, Professor Massimo Bertinaria in the University of Turin, optimized the molecule 546 and published the results in 2016. And more recently, a group in China, also starting from the hit compound 745, generated different analogs that actually are dual inhibitors of DNA methyltransferases and also HDACs. Now, more recently, we have been selecting additional small molecules and testing the activity in enzymatic inhibition assays. Last year, we identified a couple of compounds approved for diabetes, glyburide and panavinostat, here in panel A. The two compounds show kind of moderate inhibition of DNMT1 with IC50, so 55 and 76 micromolar. This project, another polyphenol compound, thiaflavine, that is a main component of green tea, also show inhibition of DNMT1 with an IC50 or 85 micromolar. The other small molecules shown in this slide are synthetic compounds from commercial libraries. And in collaboration with Professor Cathy at the Arkansas State University, we're working together with her to optimize the activity of this hit compound, 404. On this project, we're using molecular docking simulations and molecular dynamic simulations to try to rationalize the molecular level, the activity of these compounds, and also the results of the modeling are being helpful to suggest modifications to the hit molecules. Now, going back to the focused libraries. So the first step to screen the focused libraries was to put together all focused libraries commercially available. This is a project of one of the students in the group, of Alexis Padilla. So he collected 11 focused libraries. After data curation, we came up with more than 53,000 small molecules. On this table are shown the number of curated compounds and the different chemical companies that are producing these chemical structures. As part of the characterization of the focused libraries, this picture shows a visual representation of chemical space using properties of pharmaceutical interest. And the take-home message of this analysis is that the 11 focused libraries have drug-like properties, and some of them, for instance, the MedChem Express library, Target Mode library, and CellChem, are more diverse than the others. Also, part of the characterization of the focused libraries is showing here. This picture is actually a constellation map. That is another way to represent the chemical space of these compound libraries. On this slide, each dot represents not only a single compound, but a series of compounds, an analog series. And the nice thing of these maps is that very rapidly we can have a sense of the type of chemical structures that are included in the focused libraries. Now, in addition, if we represent with a color, let's say blue color, the different focused libraries, we can see very clearly and very rapidly that some of them, for instance, the chemical library from chemical diversity has actually a really large diversity covering a wide region of the chemical space. And there are other focused libraries that are, for instance, here the Acinex on the left or Otava on the right, have less diversity and are populating a specific region of the chemical space. So once, also another part of the characterization of these libraries is using a novel approach developed by Professor Ramon Miranda. He's working at the University of Florida, and he came up with a novel representation of chemical space that is called chemical library networks. On this representation, each node of the network represents an entire compound library. This approach is very fast, can be done in really few seconds. And the nice feature of these chemical library networks that were proposed by Professor Ramon is that we can actually see very large collections. For instance, the zinc library that contains more than 17 million molecules. So once we characterize the compound libraries, the next step was, of course, to do the actual filtering of the chemical libraries. So starting from these more than 53,000 curated compounds, the first step in this filtering approach was to identify the most promising ones using epigenetic target profiler. This is based on machine learning. I will talk about this epigenetic target profiler in a minute. Here, the most relevant point that I like to emphasize that this filtering is very rapid. So in a matter of minutes, it's possible to select roughly 100 compounds with the best predictions for different epigenetic targets. And then this number of molecules, 119 compounds, were subject to consensus docking using Autodoc-Bina, Autodoc, and MOE. And also we here implemented an internal extended connectivity interaction features developed by Norberto Sanchez. And after this selection of docking and rescoring, finally selected 20 small molecules for enzymatic inhibition assays. The table here shows some of the chemical structure, well, the IDs of the chemical structures with the corresponding docking scores of each one. So in the experimental screening, so the first step was to test the compounds at one single concentration, 100 micromolar. From these assays, several quinolines shown here in the slide show an inhibition between 40 and 10% of inhibition. Also, quite interestingly, we identify what is called activity cliffs. So the activity cliffs are highlighted here in color. The structure here in blue is actually an inhibitor of DNMT1 with roughly like 40% of inhibition. But the compound on the right, that as you can see, comparing the chemical structure, the only difference is the absence of two methyl groups. So the absence of two methyl groups is associated with an increase in the activity of DNA methyltransferase. So this is what is called an activity cliff, because it's a really minute, really small modification in the structure, just a couple of methyl groups, that dramatically changes the activity on the enzyme. More interestingly, were a couple of quinasolines shown here in the slide. One of them inhibit DNMT1 with an IC50 of 30 nanomolar, and the other one also inhibit DNMT1 with an IC50 of 81 nanomolar. Comparing the potency of this compound with the internal control, that is SAH, turns out to be that these couple of quinasolines are roughly 10 times more potent than the control SAH, that has an IC50 of 0.34 micromolar. Also, the couple of quinasolines shown here not only inhibit DNMT1, but also shows selectivity towards DNMT1. Both of them did not inhibit DNMT3B and show moderate inhibition of DNMT3A. Doing a literature search of these chemical structures, we came up with this paper that actually both quinasolines are also inhibitors of a different epigenetic target. In this case, an epigenetic reader, G9A and A, are also quite potent. For instance, one of the hit compounds is an inhibitor of this enzyme with an IC50 or 6 nanomolar. So putting these results together, these quinasolines are dual inhibitors of DNMT1 and also of G9A. In order to investigate the mechanism of inhibition, we combine computational approaches with experimental assays. In this case, on the left is shown, very summarized, the results of molecular docking and molecular dynamics. These analyses were done by Edgar Lopez. From these computational simulations, it was concluded that the quinasolines are actually binding not with the catalytic domain, but with the regulatory domain, specifically with this portion that is called CXXC. Now, comparing the results with the experimental assays, we identified that these molecules, these quinasolines, did not inhibit, did not compete with DNA, meaning that experimentally we can say that these compounds are not binding in the catalytic domain. And we know that we need to do additional assays here, but this is a very nice starting point, not only to identify potent inhibitors, but also to come up with hypotheses on the mechanism of inhibition at the molecular level. Now, I'm going to talk about very briefly of the efforts to identify inhibitors not only of DNMT1, but also inhibitors of other epigenetic targets. And the main approach here is using machine learning. So we know that there are very large public databases like PubChem or Chemistry that contains literally millions of data points of structure activity relationships. These very large public databases are being used by the scientific community to train predictive models using artificial intelligence. So towards this direction, the first step, specifically for epigenetic compounds, was to, from Chemistry, extract the information of small molecules targeting epigenetic targets. In this table is shown part of this analysis. The full table, the full analysis is published in the reference here at the bottom of the slide. We see from this table that there are small molecules that are heavily explored to identify inhibitors, for instance, of HDACs. Specifically for HDAC1, there are more than 3,000 compounds published in Campbell. For other epigenetic targets like bromodomains or even DNA methyltransferases, the number of information is at least a bit less restricted. But I mean, the major use of this data is that these experimental structure-activity relationships, as I just mentioned earlier, can be used as a starting point to develop predictive models. And this is what we did in this work that was performed by Norberto Sanchez. He trained several machine learning models. Here is just showing a couple of slides saying that overall they have a very good precision and sensibility in terms of binary classification and also in terms of target prediction. These models were also implemented in a public web service that is called Epigenetic Target Profiler. Of course, everybody is welcome to use this server. At the bottom of the slide is shown the web address. And the idea of this web service is that the user can actually draw the chemical structure of interest for the user, get the SMILES representation, or the user can directly input the SMILES string, predict the targets, and will show a results table pretty much like this, where the user can see the chemical structure again that is wants to predict, and at the bottom is display the number and details of the epigenetic targets for which that compound is predicted to be active. Of course, there is a column here that indicates the quality of the prediction. For the details of how this server is working, all what is done in the backend is published in these couple of references. Quite interestingly, when we made public this server, we got this comment from Professor Andrei Ursu in Florida, saying that his research group tested this server, Epigenetic Target Profiler, and correctly identified one of the small molecules that his research group is working on. So nowadays, in our group, we're using this server and these predictive models to profile other compound libraries. In particular, we're profiling compound libraries from natural origin in Mexico and Latin America. There are several research groups working with natural products, and it's of the interest to identify potential compounds for different epigenetic targets. So, to summarize, we have talked about the identification of DNMT inhibitors, specifically with novel chemical scaffolds. Also, we see how these active compounds can be used as an starting points for lead optimization programs. One of the hit compounds was actually a natural product, nanomycin. The nanomycin was the first small molecule selective for DNMT3B. Also, we mentioned a couple of quinasolines that are both potent and selective inhibitors, nanomolar activity of DNMT1. And also, doing a literature search, we identified that these quinasolines are dual epigenetic target inhibitors of DNMT1 and G9A. Also, we mentioned the modeling of compounds with known activity and other epigenetic targets. The modeling of the molecules with known activity are the starting points to develop machine learning models. And these machine learning models are now implemented for the public use in the web server Epigenetic Target Profiler. As a perspective of the research, it's to predict the activity of dual inhibitors. And this work that is done in collaboration with Professors Eli Fernandez and Fernando Prieto. Specifically here, Eli Fernandez, he developed a pipeline to predict dual inhibitors, and the implementation of this pipeline applied for natural product was done by Fernando Prieto. Second perspective is the novel design. Actually, in one of the conferences yesterday, we heard the very nice talk about the novel design. So basically, we are pursuing this general strategy using mostly freely available resources for the novel design of inhibitors of different DNA methyl transferases. With that, I would like to thank again the members of the research team, the research group, DIFAKIM, that for several years have contributed to this project. Also, many thanks to the collaborators that are doing either the synthesis of potential compounds and the testing of the small molecules. Our research group is focused on computational approaches, but of course, we know that drug discovery, as we have heard in the very nice talks on this conference, is a multidisciplinary work. So many thanks to the collaborators, and also, of course, many thanks to the funding agencies that are covering the expenses of some of the software that we're using, scholarships, and actually purchasing the compounds for doing the assays, etc. With that, I'd like to stop, and thanks again to the organizing of this conference. It's really nice, although we are several miles, kilometers apart. So many thanks to join us, Mexico, India, US, different countries participating on this very nice conference. Here on the slide, I'm showing some pictures of the campus of UNAM in Mexico City. On the left-hand side is the School of Chemistry, and the center is the main library, and on the right is the School of Medicine. Very nice murals. All of you are very welcome to visit our campus in Mexico City when you have a chance. Many thanks for your attention, and also, again, many thanks for the invitation.
Thank you so much, Professor Jose, for your intellectually stimulating research talk. Let me just ask the IT team if we have some questions for Professor Jose.
No, sir.
All right. Okay. Thank you. So, Professor Jose, we once again thank you for accepting our invitation, for taking out time, and the campus photos that you have shown, they are really looking wonderful, stimulating us. Maybe few of us can maybe visit your campus soon. And you are also invited, sir, to visit our campus, D.Y. Patil University, Navi Mumbai, India.
Thank you. Looking forward.
Yeah. So the take-home message from your talk would be epigenetic drug discovery can be done using technologies like focused libraries, using computer-guided drug design, machine learning, and experimental screening to identify novel scaffolds as drugs, as has been exemplified by Professor Jose in his talk with the discovery of DNMT inhibitors. And a good thing that has come out of, two good things rather that have come out of this talk is Professor Jose has also invited us for collaborative research, and also to use his website that he and his team has developed. So thank you so much, Professor Jose, for the talk. And as a token of appreciation, we would like to present virtual bouquets to you. The first one is the orange colored bouquet, orange colored roses bouquet, and the second one is pink roses. So the orange roses, they symbolize the enthusiasm and passion that we see in your work. And the pink roses, they symbolize our gratitude and our admiration for you and your research work. Thank you so much, sir. And the e-certificate of appreciation will reach your inbox very, very soon. Thank you so
Okay.
much. Thank you so much.
Thank you.
Good day. All right. Our next speaker, Mr. Charitra, is here. And Mr. Charitra, we'll be starting with your talk at 12, 12 noon Indian Standard Time.
Sure, sir. Thank you.
Thank you. The delegates are requested to stay tuned. We'll start at 12. Good afternoon, everyone. We'll now be starting with our next talk by Mr. Charitra Gaur. Mr. Charitra?
I can hear you, sir.
Yeah, thank you. So let me briefly introduce Mr. Charitra to the entire gathering. Mr. Charitra Gaur is basically a pharmacist. He has completed his B Pharm in Pharmaceutical Sciences from MJP Rohilkhand University, Bareilly, India, and an M Pharm in Pharmaceutical Chemistry from MIT University, Noida, India. He has delivered more than 100 sales demo, especially to Indian scientific community. So that means he's expert for the demos with respect to the Indian scientific community. And then his areas of expertise include computational chemistry, scientific software, and drug designing. He has handled many significant responsibilities, and currently he is manager technical consulting at Cube Scientific Software Solutions, India. He has more than 6 years of experience in this position. And Cube Scientific Software Solutions basically is a company that looks into the ChemDraw aspects of Perkin-Elmer informatics. So basically, Mr. Charitra Gaur would be talking to us about introduction to ChemDraw version 21. And he'll be talking about the different aspects of ChemDraw, and he'll be illustrating how it is a tool, not just for drawing. So it is beyond the drawing tool. All right. So for the next 1 hour, we'll be interacting with Mr. Charitra Gaur and listening to his talk. The platform is all yours, sir. Mr. Charitra Gaur.
Thank you, sir. Okay, let me. Should I share my screen?
Yeah, sure.
Okay. Okay, I hope you all be able to see the screen.
Yes, we can see it.
Yeah, thank you. So thank you very much for giving us the opportunity to show the features and the functionality of the latest version of the ChemDraw, that is ChemDraw 21. Okay, so this is basically the introductory presentation. We will do the demonstration on the ChemDraw as well for the drawing of the larger molecule with in a quite shorter time. Okay, so ChemDraw is a drawing tool since 1985. ChemDraw brings your molecule, chemical reactions, biological entities, and the pathway from the idea to publication to unprecedented speed. What this means is basically ChemDraw is having 3 main characteristics. One is publication grade quality. So there are some journals like JMedCan, JNatural RSCs. They have certain criteria for drawing the structure for their publication. So ChemDraw is fulfilling that requirement for you. Then we have a user friendliness. So user friendliness, like ChemDraw is evolving day by day and version by version from their palm to your fingertip. So earlier you can draw the structure with your mouse, but now you can draw the structure with your keyboard. There are hotkeys that can be utilized for the drawing molecular structure, bare molecular structure or macro structure just in a few seconds. Then the third one is the chemical intelligence. So you probably know about the ChemNMR. So you can predict the NMR spectra. You can do the stoichiometric calculation, can do the analysis, you can do the chemical properties. So all these things you can do the ChemDraw as well. Not limited to these things, but there are a lot more things we will discuss in the demonstration section. Okay, what are the modules? So ChemDraw is the first one is the ChemDraw itself. So you can draw the structure, 2D structure in the ChemDraw. Then we have, you know about this Chem3D. you can retrieve the data in the 3D part, the ChemDraw. ChemFinder to manage your chemical data in the user-friendly interface. ChemOffice plus Cloud is basically the new one. Those who are new to the ChemOffice, basically ChemOffice plus Cloud is basically you can manage your chemical data within the cloud environment. So if you have your scattered data of chemicals, like if you have a Word file, if you have an Excel file, SDF file, mole file in your local drive, you can collect those data into the cloud environment, and specifically you can manage your data with a single click. ChemExcel, within the Excel sheet, you can manage your data. You can import or export the structure into STF or mole file with the ChemExcel. And then we have a Signals notebook. Signals notebook is basically the electronic lab notebook, which is there in the cloud itself. So whatever the experiment you have done in the lab, you can just manage that particular experiment or the notebook in the Signals notebook cloud environment. Okay. Now, on the basis of the availability, there are 4 packages: ChemDraw Prime, ChemDraw Professional, ChemOffice, and ChemOffice Plus Cloud Standard. Right? Let's see what is there in the ChemDraw Prime. So ChemDraw Prime includes the 2D structure drawing, right? Then we have hotkeys and the shortcuts. So as I have told you, you can draw the molecule, bigger molecule within the few seconds with the help of the keyboard. Then we have a smart copy-paste options is there. So copy-paste like Smiles in Inkai, in Pasta, right? There are Helm toolbar is also macromolecules, so you can easily copy-paste your structures, right? So all these things comes under the ChemDraw Prime. So ChemDraw Prime is quite, you can say, quite beginners tools for the drawing, or entry-level tool. Okay, now comes the ChemDraw Professional. Let's see what is there. So ChemDraw Professional, obviously, it all includes features and the functionality of the ChemDraw Prime. Then we have NMR, that is ChemNMR. Then we have a Helm toolbar. Earlier it is known as the biopolymer toolbar, but now it is Helm one. So if you can, if you would like to draw the protein structure, DNA, RNA, Helm would be helpful for you. SNS, that is structure to name and name to structure. Right? That particular automation is available in the ChemDraw Professional. 3D cleanup option. This is very, very interesting features that is included in the ChemDraw version 21. Within the 2D part of the ChemDraw, you can just clean up your structure and you would be able to visualize in the 3D background. That is there. Color highlight and the ring fill coloring option is also there. So color highlight, like if you would like to highlight some particular area of the molecule or the functional group, you can do this by color highlight option. And ring fill color means within the benzene ring or any cyclic ring, you can fill up the different colors also. Third part is the Chem 3D is also involved in the ChemDraw Professional. SciFinder and SciFinder N is also involved, so that you can search your structure or substructure if you have a access of SciFinder, as well as the reaccess. Okay, then third one is the ChemOffice. So let's see what is there in the ChemOffice. So ChemOffice, all the features and the functionality of the ChemDraw Professional. Additionally, you can save and copy the 3D printable format .3mf file, right? So you can draw the structure and you can copy that particular structure to your PowerPoint, right? So I will show you in the next slide how you can do this. Then we have a ChemACX Data Explorer. So ChemACX is the largest, you can say, chemical data provider, right? So you can just enter the gas number and retrieve the data, retrieve the structure and their vendors, their price, their physicochemical properties. So all these things you can retrieve from the ACX Data Explorer. Additionally, in the third one, we have a Google patent. So you can search your structures, substructures with the Google patent or Google Scholar. And if you can see the last one, this particular screenshot, we can probably assume that you all know this one. This is the PubChem LCSS, Lab Chemical Safety Summary. So that is also involved in the Chem Office. So you can get the protocol for the safety of the chemicals under the regulation of the EU, under the regulation of American Chemical Society. Right? So that is also included in the Chem Office. Then Chem Office plus Cloud standard includes Chem Office. Then second one is the Chem Office plus Cloud. So as I told you, you can manage your scattered chemical data within the Chem Cloud. And then we have a Signals notebook for manage your chemical lab or experiments in the cloud environment. Okay, then we have what's new. So what's new? So here, if you can see Baloxamir, Barboxyl, this is basically the antiviral drug, latest antiviral drug. And if you can see how this particular structure is copy-paste to the PowerPoint itself, right? So you can draw this particular structure. You don't have to draw this structure in the CamDraw. So if you can just go to the name to structure module, just type the name of the drug and you can get the structure. And in the second part, if you can see the highlight group of the particular groups of this particular molecule, right? So this feature is basically .3mf. You can just draw the structure and copy paste to .3mf and then paste it to the PowerPoint. Like this, if you can see, this is the ferrocene, right? And in the ferrocene, I have also included the highlighting color section. That highlight color option is also involved in the 3D model as well. Then we have a ring fill coloring, different color you can fill in the rings. Some new hotkeys like 0 on the secondary and the tertiary carbon elongated. So if you can see this one, you can see how this particular second carbon is quite elongated. So if you press 0, it will be a elongated bond. Some other hotkeys, like if you press small j or cyclopentadiene ring, you can see it can be useful for you for drawing the metal complexes. And capital J is for 5-benzene ligand. So you can see with me here, you can see that particular benzene. And if you can see here, we have a cyclic or the linear structure of the benzene. That is the toggle aromatic ring system that you can also do in the CamDraw. Right. So thank you very much. If you have any question, let me know in the info@distributor.com or charitra@distributor.com. For more information on the CamDraw, you can go with the informatics.com. Right. So let me begin with the demonstration. Let me open the CamDraw. So this is how the CamDraw looks like. Right. This is what the interface is. Now, I would like to start with the template section. So if you can see here, we have advanced CamDraw side chain, amino acid, animals, aromatics, bicycle rings, right? Now, if you can go to the new template, right? So you can also add your own template. This is not a new feature, this is the earlier feature, but some of you are not aware, so I thought it would be great if I can show you this particular feature. So let's say I would like to go with the convert name to structure, and let's say I need to have a vitamin A. So here we have A. Then in the second box, you can click, then again, let's say vitamin B12, this cyanocobalamin, and similarly go to the third one, then structure C. Similarly, go to the structure, vitamin, let's say E. A. Right. And now I would also like to add some drugs also. So similarly, go to the structure or C. Right. Propranolol. Betamethasone, steroidal, right? So if you are working on a vitamin, if you are working on any of these drugs, you would like to draw the particular molecule again and again for your project or for your application, then you can save it as a template section, go to the file, save as, and here you should save it in .ctv file format. File name like vitamin, and once you save it, if you go to the again, here you can see in the template section we have vitamin and drugs that is already saved. In this way, you can click on any of this particular structure and do the things. Right. Okay. Now, second one is the once you go to the help, here we have a shortcuts and the hotkeys. So if you can see, if you press 0, it will be a single bond. If you press 1, it will be a single bond in a different direction. 2 for carbonyl group, 3 for benzene, right, and so on. Right. So you can just go through these cheat sheet. You can take a printout, stick to your table, and later on. Okay. Just for example, let's say I begin with the single bond, and if I press O, it will be a OH, and press 1, it will be a single bond, and let's say press 2, CO. Now, what you can do is you can just drag, press Control, and then you can easily copy the structures. Select all the 3 structures, go to the object, align, left edge, right? And what you can do is you just need to delete all bonds. And then join these bonds. So here we have our structure of the glyceride. And obviously you can do the, if you press 2, it will be a double bond. If you press 3, it would be a triple bond like that. Please excuse if my structure is not correct. Sorry for that. So in this way, you can draw the bigger molecule in a quite faster manner. Then, if you are aware of the new feature that is 3D cleanup option, right? So what you can do is say over here. Okay, let me do by this way. You don't need to draw the structure if that is there in the metrolysins. So here we have what we're seeing, right? Also, what you can do is E, and by X, click on X. X is basically for a single bond. So this way, and then press small j. So this is what cyclopentadiol ring. And once you go to the structure, here we have a 3D cleanup option, or Shift plus Control plus T. You can do the multiple time because it also involves some minimization. So in this way, you can see how this is the 2D one. You can get the structure of the 3D. If you would like to add some colors, so let me try with this one. And once you go to the colors, here we have a show ring fill color option. Okay, see this one, this one. Another thing is you probably know this particular structure. If you have gone through the website, once we click the V, it will be a cyclopropane. And you can join these structures. Press V. And here, right now, there is another thing called Control J, or joining the structures. So for example, let's say if I have this structure here, and here we have. And what we can do is I just need to select this OH and keep this shift and this NH and then Control J. Right. So this thing I need to apply for this one. So here we have this one, and here we have. So first I would like to join this strip, Control J, and then strip. Okay. And this bond, the strip. G, the structure, the structure. You can also see this thing, A, and then I would like to add some colors option, color, and for rest of the green color. And similar way, we can do press shift of this bond and shift this bond, Control J, joining the atoms. select whole structure, to the structure, So this is how beautifully you can do. Now, moving to the another feature or another new feature. Let's say, let me select this vaccine, right? I would like to copy it in 3 mm, that is a ball and stick model. Once you click on the 3D model, go to the PowerPoint presentation. I am just starting a new presentation and then Control V, right? So here you can see, you can easily copy paste the structure in the PowerPoint itself. And here we have a 3D model. You can rotate likewise. You can set up the height, width, and you can also add the animation. Enable, swing, jump. Right. So this is how this .3mf feature works. You can directly copy paste your structure into the PowerPoint, and you can do all these things. Okay. Then you probably know about the variable attachments, right? So within the same structure, you can draw multiple structures on the according to the functional group. Let's say if we have a structure like this one, or yeah. Now in this particular structure, this is rolling, so I would like to add some functional group over the ortho, meta, and the pair positions. So what I can do is, if I press shift, then go to the structure, add variable attachment, and with this I can press 2. Here we have OH. Now, if I select it, go to the structure, and generate a structure, you can see how in the different position, automated pair position, this function group has been added. So you don't need to draw the structure again and again. Another thing is square bracket magic and the round bracket. So what we can do is, okay, let's say instead of OH, I would like to add the multiple groups. Multiple groups like in the square bracket. So the square bracket, I would like to add the H2, Cl, OH, Br, and so on. Right? Similar way with the structure and then expand generic structure. So, oh, sorry, this is something which should be there. Yeah, I think this is something. Okay. So, what is the square bracket means is once you define the square bracket and the structures or the functional groups within the square bracket, the multiple functional groups will be added to the structures. We also write this way. If I have 7, 8, or cycloctane ring, and within this particular ring, if I would like to, let's say this particular ring, I would like to extend the ring. So what we need to do is we round bracket, CH2, the range of 2 to 4. Same way, and structure, right? So rings can also be extended, right? You can also include the multiple variables. Instead of single variable, you can do the double variable. Let's say we have which here, and for this and this portion, I would like to add my attachment, and for this another multiple variable attachment, and here I can define a different functional group like R over here and G3 over here. Similarly, just select to the structure, expand general structure, right? So this way you can draw a whole range of structures. If you would like to add the highlights, so the colors, select colors. Structure, right? This way, the highlight color is also working with the variable attachment structures, right? So now, another thing is the LCSS, right? So if I have some tool like we have, okay, like here, which here, so this is what fluoric acid. I would like to know all the safety summaries about this particular chemical. So once you go to the add-in, here we have a PubChem LCSS. So once you click on this, it will route to the PubChem GHS to LCSS. So you can see whatever the safety summaries under the EU regulations, European Chemical Agency regulations, SCIS regulation, just GB regulation. So all these things are there. The safety summaries. You can copy this particular statement. You can copy the precautionary statement quotes as well. Similar way, let's say we have a gas number also. So once you go to the CAM ACX Explorer, or we have another thing called CAMACX.com structure from gas registry number. So if you have gas registry number, what you can do is you can just need to enter that particular gas number, and then it will retrieve the structure. So let's say 2254 86 0. So I think, yeah, here we have a calcium orotate, structure of calcium orotate. Let's try that one time. See, we have 690-6 3. Here we have a fusidic acid. Can get the structure with this gas numbering. Also, if you would like to get the information about the vendor, the price, the physicochemical properties, what you can do is go to the ACX Explorer, and here you can see all the detail. So first you should search the exact structure, and here you can see sources, properties, sources in terms of the supplies, size, price, purity, and the catalog. If you click a particular vendor, it will route you to the vendor's website that you can easily order your chemical. Please as well. Okay. Another thing what Camp Office allow you to do is you can manage your add-in, right? So if you are working with the add-ins CamDraw, add-ins in the CamDraw, so you can make your own add-in. Like here I have my own CamDraw menu viewer, so you can see this is how we have done in the HTML format, or then Python there. So yeah, you can do RSC. I would like to know where RSC document is. So just go to the file, open style sheet, file, style sheet, and here we have a RSC document. So here you can see different journals. So if you click on the particular journal format and draw the structure, it will be compatible with that particular journal. Open sample. So here we have some biological pathways, HIV infection pathway, human insulin pathway, and here you also get the magic hotkeys cheat sheet as well. So there are some other pathways as well. That was a predefined pathway. If you would like to edit these pathways, just copy and paste to the new document, and you would be able to. Yeah. A Google patent. So if you just select this particular thing and then go to the search Google Scholar or patent, and then you can search, and by the smile code, it will get all the data. Google Scholar as well as the Google patent. Same thing. So another thing is the search with the SciFinder. If you have access of the SciFinder, you can structure of the substructure and get the data. Okay. Now another thing is the catalytic reaction. Right? So you probably know about how to draw the cyclic catalytic reactions. Right? So once you select the arrow, space, and then make it 180 degrees. Just drag the tip, press space, and then just increase the size. Go to drag the tip, and then some reactant intermediates. What you can do now is select the arrow again, just copy it and drag it back, then drag the tip. In a similar way, just select the arrow again, copy, and then drag it back. Right. So in this way, you can just draw the various cyclic reactions. Another thing I would like to let you know is the drawing organic salts. Right? Let's say we have a sodium benzoate. So what I can do is here we have Na, right? And then Control R, and then 90 degrees. Yes. For this one, so which, and if you would like to add the charge, just simply click on negative or positive on your keyboard. For this one, positive. Now, if you select these two things, right, and once you right click, go to the analysis and molecular weight. So it will show you the different molecular weight, right? Now, what you need to do is you need to group these two fragments. So what you can do is you just need to select it, go to the group and the group. Now, if you go to the analysis, molecular weight, it will show you the total molecular weight of these salts, right? Another thing is the polymer option. So let's say I need to polypeptide, I need to draw the polypeptide. So we have NH over here, and we go back this way. And once you draw this particular thing, then go to the bracket, and under the bracket, you need to label the repeat count, right? Enter a label or repeat count. So let's say we have 10 repeat counts. I'm sorry. This should be like this one. Picture. Actually, I have done this before, but I don't know why. Okay, let me try it this way. I have some predefined structures over here. Yeah, over here, like this one. So once you do this, go to the generic structures, right? This one, you can define the repetition unit like 10, and then able to get the whole structures. This, then the generic structure, right? Okay. Moving to the Chem NMR. So once you go to the file, here we have a preferences, and under the Chem NMR, you have a solvent, CDCl3, and the DMSO, and the frequency ranging from 60 to 1000 megahertz. If I can get some example for you. Right. So if you can see this structure over here, this is the 80 megahertz, and you can see this over here. How this peak is quite broad, and this is basically the 80 megahertz, under the 80 megahertz. And in the second one, here you can see 300 megahertz. Now this switch peak is quite sharp. The same way, if you go to the 600 megahertz, it is getting sharper. 100 megahertz. And like this, you can simulate your whole NMR table. Okay, so this is what some hidden feature, or you can say some familiar feature for you. Last feature is the Hill monomer toolbar. Oh, sorry for this one. So what Hill toolbar is, basically it is a biopolymer toolbar. So here, if you can see amino acids, various amino acids are there: alanine, histidine, leucine, lysine. Right. Now, what you can do is you can just need to select this, right click, go to the atom, and then expand label. You will get the structure. So you don't need to draw the whole structure. So similarly for RNA, we have thymine, right? And then right click, go to the atom, and expand label. So this is all about the features of the ChemDraw 2D part. Now moving to the ChemDraw cloud, right? So we still have some 15 or 20 minutes. What we can do is we can go to the ChemDraw cloud here. So this particular thing is to you first need to log in your Signals notebook. Right? Same credential would be there. Signals one. Right. So now. So this is what the interface of the ChemOffice cloud. From here, go to the folder. You can get the folder of various data, like if you have a Word file, if you have Excel file, if you have STF file, mol file, all the files has been scattered. You can take that particular scatter file into the one place, that is a ChemOffice one. Let's say, so here we have a bunch of STF file, right? File. Now, similarly, if we have a Word file, then it will be a Word file, Excel file, like that one. Now, once we click on the some of the structures, let's say these 4 structures, and then we can add to new collection, say ZT, and then create it. And under the collection, you can see here we have a BZ2, and those 4 structures that we have copied in this particular collection. So what you can do is you can also add the properties as well, or deselect or select properties, right, from here itself. Now, there's another thing is once you select your structures, then we have export. What export? If you would like to save it in .stf file, or PowerPoint structure report. So look, it looks like this. Right? You can define the layout, 3 rows or 4 columns, or the structure below, above, left, or right of the structures. Right? What chemical properties would you like to add? So accordingly, you can just need to click on that and then export. It will ask you for save your file. So we set T and export. 4 items were exported to bzt.ppt. Let me open. Right. So here you can see. So if you have thousands of structures, just get those files in Chemtro Cloud, and also you can search. So here we have a search option. So search by drawing, you can draw the structures and then search by creating, last time edited. So accordingly, you can search those structures. Okay. Then this is what the Chemtro Cloud says. And then we have a Seamless Notebook. Now, Seamless Notebook is basically the lab notebook, lab ELN notebook. So once you go to the, yeah. So you have your own credentials, login password, right? And this is how the dashboard of Seamless Standard looks like. Recent additions or recent experiment, awaited for review, favorite, rejected, in review. Right? So what is the workflow of this one? So once you go to the notebook, first you need to add some notebook. Like let's say drug inhibition. If you would like to add any description, you can create the notebook, and under this notebook, I need to add experiment. Experiment, like if you would like to name of is, right, create the experiment. Now, here under the content, you can see you can add the text content, chemical drawing, Word file, Excel file, PowerPoint file, material tables, sample tables, image files, right? So all these things you can do with the notebook. You can also drag and drop the files. So what you can do is, what I'm going to do is, I just need to, let's say I have this files, right? Calibration Excel file as well. TLC file as well. And then I just need to drag and drop, and you can see how this is showing. TLC, for TLC, you can just click from your mobile photo and then upload it to the Seamless Notebook. Here we have Excel file as well. Image annotation is also there. So let's say if I have this one spot, and if you like to add any one, if you add any text, it will be a tag now. Then you can enter descriptions as well. Okay. Right. So another thing is the once you go to the ChemACX, ChemACX is also there for you to help. Right. Then we have a sharing option. So you can make the group of your colleagues and then share this particular experiment to your colleagues, and then you can work collaboratively. Make sure those are also the user of Chemistry signals notebook. So, then you can sign the experiment. You can sign and add reviewer, invite the reviewer. You can sign and close the experiment as well. Once you sign and close, ask you for some declaration or digital signature. Once you click on this, sign and close, that particular experiment will be closed. That means it will be become a read-only file. I'm not going to do that, but I would like to. I would like to see this one. Now, once I share my experiment, here you can see if the class 1 members can read this particular experiment or write the experiment, that means editable form, or if you would like to give them a full control. Right? So accordingly, you can choose, and then that particular experiment can be shared. If you would not like to share the experiment, then press recycle bin or delete. History part. So whatever I have done for this particular experiment, that is saved in the history part, right? And you can also compare with the previous one. Yeah. So this is what CFR Part 11 compliance software is. And after that, once you go to the action, here we have a print to PDF. So it will save this particular experiment in your local drive in the PDF file format. It will ask you for what you need to add. I need to add history as well, and media, A4, A3, whatever the customization you can do, and then print to PDF. I have already done this, so because save the time. So here you can see whole experiment here, purpose, sample, CDXML file. And this particular thing, like Excel file or Word file, you can find it in the attachment. So you can directly download from there. So, okay, we have still 2 or 3 minutes. And the last thing I would like to show you is the Excel. So once I open the Excel, Microsoft Excel, now here you can see Chem Office 21 tab is there, right? So what I can do is I can save or manage my chemical data. So let's say we have Chem Office, new worksheet. Size. And over here, let's say we have structure. Here we have a melting point. Here we have a boiling point. Here we have a IUPAC number, IUPAC naming. So, no. First of all, citric acid. Let's say form. Right, let's say I can select these text and go to the convert and into molecule. Right? So here within the Excel sheet, I can get the whole properties as well. For melting point, boiling point, we have something called ChemDraw functions. So what you can do is under the ChemDraw functions, we have ChemPro functions, we have ChemProp standard function, according to the ChemDraw professional and ChemOffice one, you can get these functions. So let's say in melting point, we need to define a cell that is A2. Okay, and then this one. For the boiling point, 2, and then this one. For opaque, we have name, 2, and yeah. Right, so this way you can save, you can also import the structure, import like SDF file or mol file, or export the table to the SDF file. So you can also synchronize the data with the ChemFinders. So yeah, in this way you can manage your Excel data, right? So this is all about the ChemDraw. So if you have any questions, just let me know, or you can contact us to info@distributor.com or charitra@distributor.com as well. Yeah. So thank you very much.
Thank you, Mr. Charitra, for your simple, elaborate, and amazing talk. So the take-home message would be ChemDraw software is a user-friendly chemical drawing tool, which brings structures from idea to the actual drawing for publications and presentations. And you have elaborated on the different aspects, the different additions that have been made to ChemDraw. It was amazing, simply. All right. So thank you so much for the presentation, and let us just proceed on to the question answer session. Let me just check if we have some questions in the YouTube chat box. Yeah, we have one question from Dr. Anandu KR. He's asking, can you load third-party templates in ChemDraw and any database like that if it is available?
Well, ChemDraw is having their own database, CAST one. So, but uploading the template is possible, but uploading the database is, it can be done through ChemJS, that is a JavaScript, but I'm not sure that what kind of database you want. But yeah, template can be.
Okay, thank you. I hope, Dr. Anandu, that solves your query. So that's it. We had that one question in the chat box. So thank you so much, Mr. Charitra, once again. And I request the IT team to please display the presentation. All right. Thank you so much, Mr. Charitra. And it was wonderful listening from you. And as a token of appreciation, we present to you virtual bouquets of pink and orange roses. So the orange roses, they are a symbol of your enthusiasm and passion in your work. And the pink roses, they are a symbol of our gratitude and admiration for your work. Thank you so much. And the certificate of appreciation as well, I'll make sure that it reaches your inbox as early as possible. Thank you once again for that wonderful talk.
Thank you. Thank you very much. Thank you very much all.
Okay. Have a nice day, Mr. Charitra. All right, to all the delegates, we'll break for lunch now and we'll be back at 2:30 with the last two talks of this conference. Thank you. Hello, Dr. Andrea.
Hi, can you hear me?
Yeah, I can hear you. So shall we start?
Okay,
Okay, just give me a moment. I'll introduce you to the delegates.
okay.
So good afternoon, delegates, and greetings, Dr. Andrea. We are so very proud to have you here as our speaker, as our resource person. Let me introduce briefly Dr. Andrea to the gathering. He did his MSc in Pharmaceutical Biotechnologies from University of Perugia, Italy, and he has completed his PhD again in Pharmaceutical Sciences from the same university, University of Perugia, Italy. Further to his education, he has received training in as a postdoctoral fellow, which has been granted by Fondazione Umberto Veronesi at the Department of Pharmaceutical Sciences, University of Perugia, Italy. His fields of specialization include application development of medicinal chemistry approaches towards discovery, that is, hit identification and hit to lead optimization of new compounds of pharmaceutical interest, such as antiprions agent, kinase inhibitors, Staphylococcus aureus, NOR-A efflux pump inhibitors, dengue NS5 RNA-dependent RNA polymerase inhibitors. His contributions to all of these projects include application or development of different medicinal chemistry approaches, such as chemical library design, in silico strategies to rationally discover new potent and safe bioactive compounds, and physicochemical and ADMET tox profile prediction. He has received many awards and honors. His PhD thesis work has received National Award as Best PhD Thesis in Medicinal Chemistry, organized by SCI Italy, as well as an International Paul Ehrlich Medchem Euro PhD Network Certificate for Excellence. He is currently working as an assistant professor at the Department of Pharmaceutical Sciences, University of Perugia. He has published more than 20 papers till date, and his current H index is 9, and I-10 index is also 9, with around 209 citations to his work. We are indeed very privileged to have you here, Dr. Andrea, and Dr. Andrea will be talking on rational discovery of non-conventional small molecule degraders to treat neurodegenerative diseases. So over to you, Dr. Andrea. The platform is all over yours.
Can you activate my share condition, share screen?
Yeah, you can share screen.
Okay, thank you. I'm unable to share my screen. I'm not the permission.
Okay, just give me a moment.
Yeah.
Just give me 2-3 minutes. I'll just be back.
Yeah, sure.
Dr. Andrea, can you hear me?
Okay, yeah. Now it works.
Yeah, can you please try sharing the screen now?
Okay, can you see my screen? Okay, can you see my screen?
Yes, yes, we can see it.
Perfect.
Thank you.
Okay. Well, good morning to everybody, and thank you for your kind introduction, and many thanks to the organizer committee to invite me in this exciting contest. I really enjoyed all the presentation, all the talks of this today's conference. Well, thank you. As the title of my presentation said, in this talk I would like to present and to introduce you a novel drug discovery approach to identify a new class of small molecule degraders acting through a non-conventional way, and how we apply this approach to tackle neurodegenerative disorders, and in particular to target prion protein. This is a brief overview of the talk. Thus, before starting with presentation of this approach, I would like to introduce and refresh you some general information about the targeted protein degradation concept and the advantages and disadvantages of the state-of-the-art approaches to reach an effective targeted protein degradation. Then we will move on the description of this novel approach that we are proposing, and why we apply this approach to target prion protein. Finally, I would like to share with you the exciting results that we obtained within this research. Well, targeted protein degradation is an emerging and powerful tool to target disease-causing proteins difficult to hit with traditional small molecule inhibitors. The approach is based on inducing protein degradation by using a specific molecule called degrader that promote the selectivity redirection of the bound protein, in this case protein A, to degradation. Very interestingly, the state-of-the-art approaches produce this effect by coupling the binding event of a degrader to a protein A in this picture, with the modification of the molecular surface of the bound protein. This protein shape modification led to the formation of a novel protein-protein interaction surface that enables the recruitment of a second protein, protein B. In other words, the small molecule creates a novel specific surface for a novel protein-protein interaction. Well, one of the two proteic actors of this ternary complex is generally involved in cellular pathways for protein degradation. The final aim of this interaction is thus the degradation of the targeted protein by hijacking the targeted protein either to the ubiquitin proteasome system or the autophagy lysosomal system. Of note, these mechanisms provide some advantages with respect to traditional drug discovery approaches based on the traditional protein inhibitors. As first, for degrader molecules, any available pocket of a given protein can be potentially used as a target site. In fact, in conventional binary approach, in which we use small molecule inhibitors, we need to target a functional binding site of the protein of interest to block the activity of the protein. For example, for an enzyme, we need to target the active site, and thus we need to compete with an endogenous substrate of this enzyme. For example, in kinase, we can target the ATP binding site. By contrast, by using degrader molecules, any solvent accessible site can be used. In this context, we overcome the challenge of identifying suitable binding pockets able to modify the protein activity, which made it difficult to target a variety of proteins that are now described as undruggable due to the absence of activity-modifying druggable pockets. The second main advantage of degraders is that is related to their mechanism of action. In fact, the efficacy of this molecule does not strictly depend on the residence time that the molecule spent into the binding site, but only on the ability of the degrader to promote the formation of the ternary complex. Additionally, a single molecule can lead to the degradation of multiple proteins instead of act on a single protein per time. Well, generally, depending on the structure of the compound of the degrader and on their mode of action, we can distinguish between two main classes of degraders, called monomeric degraders and chimeric degraders. Monomeric degraders are basically small molecules able to induce a protein degradation. The first option for a monomeric degrader is to bind the protein of the protein of interest and in such a way promote its degradation. One of the most famous examples of this kind of molecules is fulvestrant. Fulvestrant is a selective estrogen receptor degrader approved by FDA for therapeutic use in ER-positive metastatic breast carcinomas. Even though this compound is still approved, little is known about the mechanism of action used by this compound to mediate the target degradation. The second option for a monomeric degrader rely on the interaction with the E3 ubiquitin ligase. We need to spend a bit more time on this second part, this second type of monomeric degraders. The E3 ubiquitin ligase is a key actor of the ubiquitin proteasome system. That is one of the main pathways for protein degradation inside the cells, given that it's the E3 ubiquitin ligase complex recognize and bound protein that need to be degraded and mediate the attachment of ubiquitin tags to this protein. The ubiquitination of the protein mediate a cellular signal that send the ubiquitinated protein to degradation. In this context, acting on E3 ligase are able to induce neo-substrate recognition by E3 ubiquitin ligase. In these cases, binding of a monomeric molecule to the E3 ligase resulted in enhancement affinity to proteic substrates that are subsequently ubiquitinated and degraded. One example of this mechanism of action, one molecule this mechanism of action is lenalidomide. Lenalidomide is a medication currently used to treat multiple myeloma. The binding of lenalidomide to ER3 ligase modifies the surface of E3 ligase, producing and produce a neomorphic surface able to recognize the CK1 alpha. This recognition led to the ubiquitination of CK1 alpha and its degradation. The significant advantage of, while the fact that this class of compounds are small molecules ensure a certain drug likeness of these molecules, the significant disadvantage of monomeric degraders is that they are difficult to find, and almost all have been discovered serendipitously. Additionally, monomeric degraders are found to bind either weakly to one of the two proteins individually. Well, the second class of protein degrader are the chimeric degrader. In this chimeric degrader follow those that is called ambivalent approach. Ambivalent approach to protein degradation contemplates two-headed small molecules that resume both the abilities of the monovalent degraders. Indeed, one part of the molecule corresponds to the ligand with the high affinity for the protein of interest. The other part is responsible for the recognition of the E3 ligase. These two molecules are connected by a linker to create a unique molecular entities. A unique molecular entities referred to chimeric degraders. One of the most famous chimeric degraders are the products molecules. The key advantages is that the fact that bifunctionalities allows exchange of the target binding warhead in a plug-and-play fashion to interactive change the specificity of the target of the final molecule. However, this modularity at the same time also gives rise to a key shortcoming, as it renders the compound comparatively large and make it more challenging to progress in terms of ADME properties. In particular, these molecules can encounter problems with toxicology, with uncertain toxicology, or the adverse effect of these kind of molecules is still unknown. In particular, they suffer from oral bioavailability, in principle, in mainly because mainly due to their high molecular weight and large polar surfaces. Thus, even though this class of molecules is more simply for this class of molecule is more simply the application of a rational discovery program to tackle a given protein of interest, the optimization of ADME properties is more challenging. Well, in this context, we have proposed for the first time a new discovery approach for the identification of a novel class of small molecule degraders called PPA-FIT, or pharmacological protein activation by folding intermediate targeting. Well, the innovation introduced by this approach is that while conventional degraders act on the native form of targeted protein, we try to rationally design to rationally target the folding intermediate of the protein of interest. Indeed, as you know, before to acquire its native conformation, a protein undergoes to a process named protein folding. The protein folding is a process by which a polypeptide chain in its unfolded state folds to become a biological active protein with its proper native conformation, proper 3D conformation. The native structure represents the most stable structure of the polypeptide chain. But during its folding, a protein can explore one or more metastable states, also called folding intermediate. PPA-FIT aim is the identification of a small molecule targeting the folding intermediate. Why a small molecule able to target the folding intermediate? Because the binding of this small molecule to the folding intermediate led to the stabilization of the folding intermediate. and to the production of a non-completely folded structure that the cell can recognize as a non-correctly folded structure. This protein, stabilized conformation of the protein is thus sent to the protein, to the degradation pathway. And in this way, we could be able to lower the levels, the cellular levels of the targeted protein. Of note, PPA-fit degraders can share advantages from both the previously mentioned degraders. In fact, as for monovalent degraders, PPA-fit degraders are small molecules, and these particular aspects ensure a certain dark blackness of the identified compounds. In parallel, as chimeric degraders, PPA-fit degraders can be identified using a rational approach, and thus can be used for a wide range of targets. Well, in addition, the PPA-fit approach, the identification of a pocket in the folding intermediate, expands the applicability of protein degraders to those targets that do not provide a solvent-exposed druggable pocket in their native structure. Well, starting from with this idea in our mind, we decide to apply this approach to prion protein. Why prion protein? The prion protein is the causative agent of prion diseases that are a family of rare neurodegenerative disorders with a common etiology, that is the neurotoxic activity of prion aggregates. In fact, prion protein can exist in two different conformations. In the physiological conformation, also called PRPC, the prion protein is rich in alpha helix and located in the cellular membrane of the neurons. By contrast, in its pathological conformation, the PRP, the prion protein, is rich in beta sheet, called PRP scrappy, and has a high propensity to form aggregates around the neuron. These aggregates mediate a neurotoxic activity. Additionally, PRP scrappy can propagate itself in a process called prion replication. In particular, this process, it was shown that PRP scrappy interacts with PRPC, thus the physiological form, and is able to promote the conversion of PRPC in PRP scrappy form. Additionally, the interaction between PRP scrappy and PRPC can promote, is necessary to the intracellular transduction of the neurotoxic signal. As you can see, PRPC play a central role in all the neurotoxic activities of these pathologies. And for this role, and for this reason, PRPC is considered the optimal target to tackle to fight prion pathologies. In this regard, it was demonstrated that lowering PRP cellular levels is proved to have favorable therapeutic effect. Indeed, removing PRPC from the cellular membrane prevents the formation of and the propagation of the misfolded PRP scrappy form, as well as the intracellular transduction of the neurotoxic path. Additionally, it was also shown that the genetic suppression of PRP in animal models produced little or any side effect. Taking together, all these considerations explain why a lot of efforts have been made in the identification of a small molecule that prevent the PRPC-PRP scraping interaction. For example, different strategies have been applied. For example, it was tried to lowering PRP levels by blocking gene expression. It was tried to promote the relocalization of PRPC from cellular membrane to endosome and lysosomes. But also it was tried to bind and stabilize the PRPC form, preventing its the formation of the pathological PRP scleriforms. However, up to now, none of these approaches have been proved to be effective, and no small molecule with an effective therapeutic effect on prion disease have been identified. In this context, is emblematic the work performed by Reidenbach and co-workers. The authors used 4 different screening modalities aimed at discovering binders of the human prion protein. They used both experimental screening, in vitro screening, and in silico screenings to evaluate the ability of a huge amount of compound to bind the prion protein. Despite the large number of molecules tested and the complementarity of the used approach, they were unable to identify any hits suitable for advancement into drug discovery programs. They only obtained partially positive results from a fragment screening campaign with the identification of 1 fragment and several analogs that have a poor affinity for PRPC. We are talking of a KD higher than 1 millimolar, and no improved binding of chemical analogs were found, and this precluded their final validation as positive hits. The authors finally conclude that observed low hit rate suggested that PRP is a difficult target for small molecule binders, probably due to the absence of obvious binding pockets on PRP native structure. However, it is true for the application of traditional approaches. In fact, in this regard, it was experimentally reported that the prion protein explore a folding intermediate along its pathway. And the presence of a folding intermediate and the therapeutic benefit showed by lowering PRPC cellular levels make the targeting of this protein, prion protein, a perfect proof of concept target to test PPI-fit approach. Well, in this context, we decide to apply for the first time the PPI-fit approach for the identification of the first PPI-fit degrader, in principle able to bind and stabilize the folding intermediate explored during the folding pathway of prion protein, and to send the stabilized folding intermediate to protein degradation. Well, to apply this approach, we build and develop a workflow that was the results of the cooperation of different groups and disciplines that integrates and that were integrated in a unique workflow and in a step-by-step results. This integration allowed the progression of the project from the identification of folding intermediate from the reconstruction of the folding pathway to the final biological validation. Well, the first strong computational efforts was the reconstruction of the prion folding pathway. Starting from an initial thermal denaturation of the prion protein structure, 118 refolding trajectories were collected and used to reconstruct the folding pathway. This work revealed the presence of an on-pathway PRP folding intermediate in the near proximity of the native states. The intermediate conformation was very close to the native structure, and in particular, the intermediate was characterized by a displacement of the helix 1 PRP with respect to the native form. A little consideration about the lifetime of this folding intermediate is needed. In fact, we need to keep in mind the following consideration. It is widely reported that the folding kinetics of the vast majority of the proteins in vitro ranging from millisecond to second scale. Additionally, we need to consider that longer time are required itself because of the ribosomal progressive translation process, which reached the maximum rate of 10 residues per second in eukaryotes. Therefore, the identified folding intermediate is likely to exist for an amount of time well over the binding rate of a classical binder, which is typically estimated to occur in a millisecond. Once identified folding intermediate, we scouted its surface in searching for a druggable pocket. And to do this, we applied a combination of MD simulations and protein scouting. In particular, we paid particular attention on the regions created by the displacement of helix 1, because a pocket in this region can ensure a pocket present in the intermediate structure and not in the native structure. Finally, we selected a pocket placed between the alpha helix 1 and alpha helix 3. This pocket was defined by residues that in the native structure are not solvent exposable. As you can see, the druggability of the pocket was evaluated using two independent softwares, sitemap and DocZ-Scorer. Well, in terms of hydrophobic and hydrophilic properties of the pocket, the identified cavity was characterized by an hydrophobic core, an hydrophobic region placed at the center. That is the region that was then occupied by hydrophobic residues in the following folding steps. This hydrophobic core was surrounded by the hydrophilic region. I want to point out that both components, both the hydrophobic and hydrophilic components, are essential to define the site as druggable. Indeed, an important parameter that we considered was the balance, that is the ratio between hydrophobic and hydrophilic surface of the pocket. Well, The obtained pocket, identified pocket, was then submitted to virtual screening process of a commercial library. Indeed, thanks to the fact that PPA fit degraders consist on small molecules, we can select a traditional commercial library for our virtual screening workflow. Each compound was docked within the selected binding site, and we generated 10 poses for each docked compounds, then submitted to a rescoring step. Only the best rescored pose was retained, and finally the results were filtered based on pharmacodynamic and pharmacokinetic properties. In fact, given that our final aim was the degradation of the prion protein, protein expressed in the central nervous system, we removed, filtered out the compounds with an inappropriate ADMET profile. That is one of the main issues in drug discovery steps following the hit identification. Well, at the end, 30 compounds were selected and purchased for biological validation. The final selected 13 hits. For the final selection, we used a diverse-based approach. In this way, we optimized the explored chemical space in relation to the number of purchased compounds. These 13 compounds were sent to biological validation. Well, the first step, the primary screening that we performed, was based on the ability of each one of these compounds to lower the amount of PRP protein in cellular systems. We tested four different concentrations, and we considered as a positive hit a molecule able to lowering the amount of cellular PRP at least at 13% with respect to the control. Well, of the 30 compounds, 4 turn out to be actually able to lower PRP levels. And I will point out that as these compounds share the different scaffolds, underlying the ability of the applied workflow to select different chemical diverse molecules that actually showed a biological activity. In particular, one of them, SM875, hereafter called SM compound, showed a very interesting EC50 in a low micromolar range. Thus, further biological characterization was performed for this compound. As first, we observed that SM compound is able to lower PRP cellular levels in a dose-dependent response manner and in different cellular models. For example, are depicted the results obtained for X cells and for ZR cells. Well, while X cells are transfected cells in which the PRP are inserted through transfection, ZR express prion protein in an endogenous way. In both cases, we observed a dose-dependent curve, ruling out possible artifacts that can be obtained in transfected cell lines. Then we observed a selective effect of PRP. Indeed, the same lowering effect obtained for PRP was not observed for NEGR1 and T1. NEGR1 and T1 are two GPI anchored protein, as PRP is, that following the same biosynthetic pathway of PRP. The information obtained by these results is that SM compounds have a specific effect on PRP levels, and that this compound does not affect in a specific way the intracellular protein traffic. Then we found that SM compound lowers cellular levels of prion protein, but have no effect on the level of prion mRNA. In fact, real-time PCR was used to assess the level of PRP mRNA in treated cells, and the obtained results are in line with the proposed mechanism of action. In fact, SM compound does not seem to have any relevant impact on PRP mRNA, regardless the cellular line. Then we also rule out the possibility that SM compound can lower PRP levels, acting on PRP native form. To demonstrate this evidence, we perform a DMR, dynamics mass redistribution experiments, a biophysical technique previously employed to characterize small molecule ligands or binders of PRP. Well, by contrast to tetrapyrrole, that is a known PRP binder, no detectable binding was observed for SM-875, neither for human or mouse form of PRP up to 100 micromolar, excluding any possible interaction of SM-875 with the PRP native structure. Additionally, we observe also that SM compound act only in the presence of the nascent PRP chain. To obtain this information, we use a cellular system in which the prion expression is regulated in the deoxycycline inducible manner. If the cells are exposed to deoxycycline and thus to the PRP transcription is activated, in the cells coexist both the native form of PRP and the nascent form of PRP. In this case, SM promotes the cleavage of PRP, as shown by Western blot results. By contrast, if the deoxycycline was removed, and thus we in the cell have not nascent PRP chains, no cleavage was found mediated by SM compound. Well, finally, the ultimate aim of an anti-prion compound is the ability to abrogate prion replication. Encouraged by the collected data, we tested the ability of SM compound to inhibit the replication of prion strains in infected mouse fibroblasts. Well, we found that SM compound inhibit prion replication in a dose-dependent fashion, also in this case, but very interesting, in a similar way to tetrapyrrole, that is one of the most potent anti-prion compounds known up to date. Well, in conclusion, herein I presented a novel approach to target protein degradation called PPI-FIT. The application of PPI-FIT technology allowed the identification of SM-875 as a novel small molecule degrader, unconventional degrader, with a promising cellular activity. all the collected data are in agreement with the proposed mechanism of action of SM875 as PRP folding intermediate binder. However, it is clear that we do not have the direct evidence or the experimental proof that this interaction appear. We performed several attempts to produce this kind of information using X-ray and NMR experiments, but we did not obtain a clear indication. And it is mainly due to the technical limitations that we encountered when we try to work with a folding intermediate, given its transient nature. Well, for this reason, we are exploring novel and innovative ways to obtain more direct experimental evidences of the interaction between folding intermediate and SM875. In parallel, we have already built a preliminary subprofile around the SM compound that we are expanding. And further virtual screening are ongoing to identify novel and more interesting compounds. Well, finally, I would like to thank all the people that work around this project. In these slides, I put just the main player of the project. And I would underline that the PPA Fit is a technology exploited by Sibylla Biotech, a startup funded by some of the main players of this project. Well, thank you for your attention, and if you have any question, I'm...
Thank you, Dr. Andrea, for that interesting and thought-provoking research talk. Let me just ask the IT team if you have questions in the YouTube Q&A chat box. Yeah, people are all complimenting your talk, sir. Most of them are saying that PROTACs are really interesting and the session was amazing.
Thank you.
Yeah. I have a question for you. Can this approach be used for any of the protein misfolding diseases? Like for instance,
for which kind of protein, sorry?
Misfolding diseases like Alzheimer's.
Misfolding proteins. Actually, this kind of approach acts before the protein can misfold. This approach acts on the natural folding process of the protein. Thus, the misfolding is somewhere that happens in a subsequent manner. And the aim of this approach is to simulate the misfolding of a protein and to more than to drive the recognition of an incorrect folded state as a misfolded state.
Okay. All right. Thank you. So the take-home message from your talk would be, a rational design of small molecules is possible for targeting the protein misfolding diseases, neurodegenerative diseases, by using your novel approach of non-conventional pharmacological protein inactivation by targeting the folding protein intermediates. So thank you so much for that talk. And let me quickly ask the IT team to share the slide. Yeah. So thank you so much, Dr. Andrea,
You too.
for accepting our invitation and being a speaker for our international conference on advances in pharmaceutical medicine chemistry 2022. And thank you so very much for your interesting talk.
Thank you too.
Of appreciation, the e-certificate will reach your inbox very, very soon. Thank you so much.
Thank you. Thank you.
Have a nice time. Bye-bye.
Bye.
Hello, ma'am. Good afternoon.
Good afternoon.
How are you, ma'am?
I'm good. Thank you.
Okay. We'll just start in 10 minutes, the scheduled time. Okay.
Yeah, sure.
Thank you, ma'am.
So can I start my presentation or you want to display something else before that?
Yeah. I'll be displaying your bio.
Okay.
Yeah. So,
No, I thought you're doing it after, so I was wondering.
okay. Thank you, ma'am.
Okay, fine. So yeah, I'll get back in a few minutes.
Yeah, sure, ma'am.
Okay.
Thank you.
I'm here only, but I just
Okay.
So Harish, if I may ask,
Good afternoon, everyone. Good afternoon, respected speaker, ma'am, and to all the delegates. So now we are towards the end of our 2 days international conference, and let us all welcome our valedictory address speaker, Professor Mariam S. Degani. I feel immensely proud, and it is my immense pleasure to invite you, ma'am, to the dais, to the online dais. Let me introduce Professor Mariam Degani. This is just a short bio of her. Her resume extends so many pages. I had to really look into all the aspects to get into the small resume, to make a small resume out of it, small bio out of it. So her education is B.Pharm in Pharmaceutical Sciences from University of Mumbai, M.Pharm in Pharmaceutical Chemistry from University of Mumbai. PhD she has completed in Pharmaceutical Chemistry from SNDT University, India. And her fields of specialization include 2 broad areas. The first one is drug discovery and the second one is process chemistry. In drug discovery, she is involved in computer-assisted rational drug design and the synthesis of small focused libraries of therapeutic agents for tuberculosis, Alzheimer's disease, and cancer. In process chemistry, she has been involved in fluorine chemistry, deuterium chemistry, task-specific ionic liquids, and flow chemistry-based process optimization. She is also actively involved in various industry and consultancy projects in the areas of process chemistry and drug discovery. She has received many awards and honors. She has received a Distinguished Alumni Award by CU Shah College of Pharmacy, SNDT University. She has been a recipient of the Ghada Award for Research Publications. She has received the Best Teacher Award of ICT for 2 times. She is a fellow of Maharashtra Academy of Sciences. She has held many significant responsibilities in the past. She has worked as a reader and HOD in ICT, Mumbai. Previously, she has worked as a senior lecturer in SNDT University, Mumbai, India. And currently, she is working as Sir Dorabji Tata, Professor in Pharmaceutical Chemistry at the Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai, India. She has guided more than 28 PhD students, and that includes me as well, and more than 75 master's students. Her publications and presentations, she has published more than 90 peer-reviewed national and international papers. She has 2 international patents and many Indian patents to her name. She has authored 1 book, and she has delivered many invited talks in various national, international conferences and symposia. Her current H-index is 21, I-10 index is 49, and her publications have garnered more than 1,500 citations. So we are indeed very proud, ma'am, to have you here as our valedictory address speaker. And Professor Mariam Degani would be talking to us over the next half hour for nitroheteroaromatics for tuberculosis, the challenges and potentials. Welcome, ma'am, and the online dais is all yours.
Thank you very much, Dr. Kundaikar, for that very kind introduction. It's been my pleasure to be associated with you both in the past as well as I'm happy to associate in a new capacity right now. So today I'm going to talk a little bit about some of the research sharings that I have regarding a class of compounds which can be a boon or a bane. What I'm talking about is nitroheteroaromatics, which can be toxic or they can be good for many diseases. So I'm going to try and share the screen now. I'm not seeing that.
Can see your screen, ma'am.
Okay, fine. You can still see the screen?
No, ma'am, not now.
Yeah. Yes, so here we are.
Yeah.
The topic for today's short talk from me is nitroheteroaromatics for tuberculosis, both the challenges and the potential. And to begin with, though I'm sure many of you already know this, I'm just going to do a little bit of the significance of the infection Mycobacterium tuberculosis. As early as 1882, Robert Koch said 1/7 of all human beings die of tuberculosis. This was almost 150 years back. Unfortunately, even today, the death from tuberculosis is quite high, though not as much as before. In 2020, an estimated 1.5 million people died from tuberculosis, including those with HIV as a co-infection. However, drug-resistant tuberculosis remains as a major public health crisis. And unfortunately for India, the statistics are not so good. They account for 26% of all these 9.9 million people who fell ill from TB in the recent years, and 27% of the drug-resistant TB. But it is not only an India-specific disease. This disease, especially resistant tuberculosis, is spreading through the world. So definitely we need to see what we can do as medicinal chemists to stop this. Let's go on to the main characteristics of Mycobacterium tuberculosis, which distinguishes it from other bacterial infections. It has a much slower growth rate, almost 10 times as slow in vitro as compared to some of the other bacteria. In addition, it has a highly lipophilic and low permeable cell wall. So that helps to protect it from drugs. It resides in the macrophages, usually in the lung cells, but also other cells. So it is intracellular in residence. And as we all know, it is resistant to antibiotics. And several of the drugs which were very useful earlier have now are no longer capable of being used for this. So MDR, that is multi-drug resistant, and XDR, that is extensively drug-resistant TB, are major concerns. In addition, there is one more thing. It can nicely sleep. So it can be latent. It can be dormant for years and years together inside the lung cells. In fact, in India, it is estimated 1/3 to half the population has latent tuberculosis. But luckily, as long as our immune system is good, it doesn't matter. However, when there is some problem with the immunity, this rears up and can be fatal. So looking quickly at the timeline, I don't want to read through this whole busy slide. I just want to share that the first drug in modern therapy was streptomycin in 1940. And then we came to rifampicin in the late 1960s. From 1960 right up to almost 2000, there was no new drug in the market because people thought this is fine, this is enough, no need to do any more research. And if you see, even this drug is rifampicin, which is just a derivative of rifampicin with better pharmacokinetics. So it's nothing very great. However, at this point of time, people realize that there is resistance developing. And though pharmaceuticals, big pharmaceuticals were not interested in doing research in this disease, many NGOs and governmental bodies came together and funded research for this. And today we have some more drugs in the market: bedaquiline, delamanid, and pritomanid. And now that brings us to the topic for today, that is delamanid and pritomanid are nitroheteroaromatics. So getting to the nitroheteroaromatics, let's understand something more about them. They usually have potent activity both in active and latent tuberculosis, and they have a unique mechanism of action as compared to all the other drugs that were listed out earlier. Therefore, there are minimal chances of resistance or cross-resistance to existing drugs. Just to give you a brief idea, these are some of the nitro-containing drugs, some of which are still very much used in the market, and some of which, like chloramphenicol, is almost outdated. So there is toxicity involved with these compounds, and there is really good therapeutic activity involved in some of the compounds. So this nitroaromatic is like a balance of both. We have to understand the type of molecule, the chemistry of the molecule very nicely before we can use it for further therapeutic purposes. One of the nitroaromatics are the nitroimidazoles, out of which we have already seen the structure of metronidazole. And these can target a broad range of parasitic and bacterial pathogens that infect different parts or different sites in the body. So there are a lot of potential uses for this. And going back again to the history, one of the first nitroimidazoles was obtained from a bacterial extract and used to kill Trichomonas vaginalis. Azomycin was the active compound from which metronidazole was developed. Metronidazole was repurposed over the years. I know you had already got a lecture on repurposing, but you can see that it's used for a wide variety of infections. It was tried in the 1990s for Mycobacterium tuberculosis, but though it was partly active, it was not really successful. But from this metronidazole came the design of newer compounds. So what are nitroimidazoles? They are prodrugs that require bioactivation of the nitro group for their antimicrobial effect. The molecules generally enter the cell through passive diffusion, where in the cell, the nitro group is reduced to a reactive radical species, which can react with cellular components such as DNA or protein. Now, it's interesting that though there are a lot of nitroheteroaromatics, they do act with slightly different mechanisms. The mechanisms all depend on the reducing capability of the nitro group, but in different ways. So, the nitroimidazole reduction products formed in the cell depend on the redox potential of the compound and the number of electrons involved, and under anaerobic conditions, the redox potential of the electron transport system in microbes is sufficiently negative to reduce the nitro group. That is not usually sufficient for reducing the nitro group in the animal cells. Under anaerobic conditions, on the other hand, the cytotoxic nitro radical is rapidly reoxidized to its parent drug in a process which is called futile cycling. And some of its bactericidal effects may be diminished. So this is a picture of futile cycling, which finally generates peroxides, and that can cause damage. You can see that there are radicals also formed which can cause damage, but the maximum damage is when toxic reduction products are produced. For example, hydroxylamine, nitroso compounds, and these can also cause damage to the cell. The electron donors involved in this process vary across different species, and that is critical in designing a molecule which is safe for humans but toxic for microbes. So what is the key thing about toxicity for nitro? In fact, for all medicinal chemists generally learn right in the beginning, nitro group is a structural alert. Don't try nitro so easily. Don't try nitro aromatics so easily. That's the tendency. Why? One, of course, it's highly electronegative and it behaves a little differently from the other electronegative groups. It has electrophilic behavior. It is very much prone to redox reactions. That is the main thing. And it can be converted to toxic intermediates. So we can see some over here, some of the possible toxic products. I'm not going to go into the details. I know it's a very busy slide. I just want to highlight that here it can form hydroxylamine, the nitroxyl radical, and the radical anion. These tend to be toxic and can damage cells. So what cells do they damage? Depending on where they are, they can damage human cells. They can cause liver problems. They can cause methemoglobinemia, bone marrow depression, allergic reactions, and they can covalently even bind to the heme and the albumin. So what has to be done is the structure of the groups next or near the heteroaromatic ring should be modified so that the toxicity is minimized. But at least for anti-infective, at least for tuberculosis, it can kill the causative organism. So one of the possibility is optimization of the electronic and stereoelectronic properties. We can see that changing groups near the nitro or even para to the nitro can modify the structure so that it is relatively safe to use. Identification of nitroactivating enzymes and understanding that so that it does not damage the animal cells. We can also think of limiting the systemic exposure by modulating the pharmacokinetic properties, simplest of which are log P, log D, pKa, etc. And understanding the genotoxicity and mutagenicity, and then modulating the structures to obtain better activity. Now, I'd briefly like to go through some of the research that we have done in this direction. Harish, please stop me if I go too long because we have done quite a lot of work and I know time is limited. So the initial development of the nitroheteroaromatic pharmacophore was designed using the data that we had already available, but we decided in our lab, instead of looking at the nitroimidazoles, which were very much explored, we looked at other simpler heteroaromatics, and we looked initially at the furan ring. Instead of having a bicyclic ring system, we looked at a simple spacer and then attached it to another ring. And in this way, we designed several compounds after looking at other people's literature. Not only that, we generated a pharmacophore model, and this was developed with an MEP map, and we understood the importance of what groups should be at what electronic positions, what electronic positions should have to have increased activity. On doing the pharmacophore modeling and density function analysis, we found out an idea not only about the possible activity, but also about the toxicity of these compounds. These are some of the analysis that we got, where we designed compounds which could have this pharmacophoric background. Thus, as I told you, this led to the design and synthesis of several molecules. Series of molecules were designed. And the pharmacophore highlighted the structural requirements important for anti-tuberculosis activity. QSAR visualized the context of different properties with one or more active or inactive ligands, helping to get a deeper insight into the activity. And not only that, the quantum chemical properties such as LUMO and electron affinity were calculated to understand better the stereoelectronic features. So with this, several compounds were designed. Without going into the details, I would say that several small libraries were designed. The set 1 compounds had activities between 1.6 and 46 micromoles. That led to the design of further modifications, and it was found that a trifluoromethoxy group led to better activity, and tertiary amine derivatives also at this position led to better activity. So these tertiary amines were part of an alicyclic ring, then they led to better activity. The moment larger groups were made, you can see that this activity decreased considerably. So here we have got a compound with a sub-micromolar activity, which could be taken further. And this had some properties. Again, the problem with some of these compounds is not only their toxicity, but also their solubility. So one of the problems was the solubility of these compounds was very poor. And to improve the solubility and reduce possible toxicity, we decided to convert the sulfur beta unsaturated ketone to a cyclized form. Again, a lot of calculations and understanding was involved. But finally, when we synthesized the compounds, we got compounds of this side. And we had also done one more modification. We had replaced the oxygen with a bioisosteric sulfur over here. And these compounds that were designed later improved the solubility and had less toxicity than the original compounds. We had also done a lot of CAD work to understand the details of this. And unfortunately for these compounds, though they improved the solubility, their activity was retained. It was not as potent as the earlier series. As I told you earlier, the mechanism of nitro compounds was reduction. And to confirm the mechanism of action of the designed compounds, we used a very simple cyclic voltammetry method. The nitro group was confirmed to be responsible for the mechanism of these, and a preliminary correlation between the electrochemical behavior and the tuberculosis activity was understood. So again, I'm going to be very brief in showing this over here, that compounds which were inactive were not having a very strong trough at position 1. If you can see, this and this were inactive, and compounds which were active had prominent troughs, and these occur at lower reduction potential values than the inactive compounds. So this correlated with the anti-tuberculosis activity that we had found. So we developed an SAR of the compounds we made, And in case of the derivatives with phenoxy linker, the nitrothiophene scaffold with the terminal phenyl substituted with electron withdrawing groups was superior to similarly substituted nitrofuran scaffold. In case of the derivatives with the benzyl oxy linker, the nitrothiophene scaffold was preferable over the, again, the nitrofuran scaffold. However, in case of the tertiary amines, the nitrofuran is the scaffold of choice. So both nitrofuran as well as nitrothiophene scaffolds have tremendous potential for developing anti-TP derivatives. Then these were evaluated, and you can see over here that three most compound of the nitrofuran series and nitrothiophene series were taken for further biological evaluation. Not only were they screened on the normal H37RV, that is the classical strain, but also against a resistant strain and against other mycobacterium species. It is interesting to note that the compounds which were quite potent in both the normal as well as the resistant species sometimes did not have any action on other mycobacteria. That means what we can conclude is it's not good to use other bacterial or other mycobacterial species because they may not have correlation with the activity that we obtained over here. So derivatives are selected towards slow-growing mycobacteria compared to fast-growing bacteria, as we can see over here. And we can also see, as far as E. coli and S. aureus were concerned, that most of the compounds were highly inactive. When we evaluated it for cytotoxicity, because again, that can be a problem, the most potent compounds had a relatively decent selectivity index. You can see that it is in the range of 50 to 100 microgram per ml as compared to the biological activity, which is less than 1 microgram per ml. And here you can see the selectivity indices. That's what I was talking about in the last slide. And we can see that for some of the compounds, these are very safe as compared to their toxic effects. Same data is for another cell line versus the mycobacteria. The intracellular infection model was also assessed for the action of these drugs, and it was found that even in these, decent activity was obtained from these compounds. Another interesting feature is we did synergy studies. Now, unfortunately, the synergy studies could not be easily done in tuberculosis because of the biosafety restrictions that were there. But in M. bovis, they were found to be additive to other known antimycobacterial agents like isoniazid, rifampicin, and ethambutol. However, a good study with the H37RV would be more useful for the synergy studies that is yet to be done. This shows a possible receptor, TBNAT, where these could possibly act. And another interesting feature of these compounds was they also acted as efflux pump inhibitors. So 3 of the compounds, 15, 16, and 17, showed much better efflux pump inhibition than the standard inhibitor verapamil. And therefore, this property could be used to design an adjuvant for even the current antimycobacterial treatment, as they would prevent efflux pump throwing away the drug and the bacterium developing resistance. Another study that was carried out was the nutrient starvation model. That means basically we're looking for its activity in latent tuberculosis. And it is found over here that against some of the compounds over here shows activity in comparison to the drugs which are currently used. So what interestingly is these molecules 21A2C, 21E, and 25, which were only moderately active in the whole cell inhibition assay, were highly potent in the starvation model, and vice versa. What it means is they are acting, some of the compounds, they are acting by two different mechanisms, one in the latent cells and one in the aerobic conditions that we had seen right in the beginning, which I had mentioned. So that can be interesting to go ahead for the mechanism, and that is shown also by the drugs which are now already in the market. They show different modes of activity in latent and active species. Thus, I want to summarize this part, that several hits were generated showing greater than 1.5 log-fold inhibition in latent tuberculosis model. And the scaffold was found to be selective towards tuberculosis, not active against other species, which is also useful because these days we want to have not a broad spectrum, but a narrow spectrum compound, especially for targeting tuberculosis. So just to summarize, this is some of the range of scaffolds which we have synthesized in the in my laboratory. And I have already mentioned the most potent compound was found to have a sub-micromolar range, but there are lots of compounds actually within the sub-micromolar range. And further work obviously has to be done for lead optimization and in-depth understanding. Though we have done a little bit about the mechanism, we need to have further understanding of the mechanism of action. With this, I would like to really thank my institute, my students, without which this work would not have been done. Of course, Harish has also done wonderful work, but in another subject. And this is my research group. I would also like to thank the organizers and the people listening in for sparing their valuable time. And I would be happy to address any questions that you may be having. Thank you.
Thank you, ma'am. It was wonderful listening to you. And the results are really impressive. Let's all hope that we get a potent anti-TB molecule from MST lab in future, in immediate future. Okay, I'll request my IT team to please check if there are questions in the chat box, YouTube chat box. All right, it seems there are no questions, ma'am. So I would like to thank you again. And the take-home message from your talk is, you have very succinctly summarized the drugs that have been available since 1943, starting with the first drug streptomycin for anti-TB activity, right up to 2020, and you have covered it in a gist. The importance or underlying importance of the significance of the nitro-heteroaromatics, and bringing out the importance of the nitro groups in conjunction with the furans or thiophenes or imidazoles or the other heteroaromatic groups for their anti-tubercular potentials. So thank you so much, ma'am, for your talk, for your valedictory talk, the last talk of this international conference, and who better than you to give us a talk, ma'am. Thank you so much for your talk on nitro-heteroaromatics. For
just like to share one more slide, if you don't mind.
Sure, ma'am. Sure. Our pleasure, ma'am.
So So I don't know if you can see my screen.
And the screen is visible, but we can still see.
I'll just.
Yeah.
Since it is the valedictory talk, I thought I can share with you. I don't know if the screen is visible still.
No, ma'am, it's not visible.
My thoughts on medicinal chemistry. It's not easy to be a medicinal chemist. It's a long way to go. Biology, maths, drug design. All this you must know. All this and more you must know. Understanding molecular mechanisms and in understanding molecular mechanisms is a must. And in drug discovery, you must collaborate or go bust. It's not easy to be a medicinal chemist. It's a long, hard road there, but my heart lies there. Thank you.
Wonderful, ma'am. Wonderful. Thank you so much. Right. May I request the IT team to share the PPT, please? Yeah. All right. So thank you so much, ma'am. And as a token of appreciation, we would like to virtually felicitate you with the virtual bouquets of orange and pink colored roses. So the orange colored roses, they symbolize the enthusiasm and passion for your research that we see in you. And the pink colored roses, they are to express our gratitude and admiration for you and your work, ma'am. Thank you so much. Thank you so very much. And as a gesture, again, as a certificate of appreciation, we would like to felicitate you with the e-certificate. And I'll ensure that it reaches your inbox very, very soon. Thank you so much, ma'am. Ma'am, may I request you to just stay around? I mean, we also have a NAC inspection going on. So I'll just quickly check if our principal sir is ready to join the validation.
Sure.
Hello, ma'am.
Hello.
Yeah, ma'am, unfortunately, principal sir is still into NAC related activities, so he won't be able to join. So I'd request you to declare the session, the scientific sessions as closed.
You want me to do that? Okay. I'm happy that you all have had a wonderful conference, and it's my pleasure to declare that the conference is closed. Thank you.
Thank you so much, ma'am. And now let me move on to the vote of thanks. So good evening to all of you, the respected speakers, Professor Mariam Degani, she is already present here, and to all the delegates. So I, on behalf on DY Patil deemed to be University School of Pharmacy, convey my deep regards and hearty thanks to our founder, Dr. DY Patil sir, our president, Shri Vijay Patil sir, and managing trustee, Mrs. Shivani Patil ma'am. I would also like to thank our convener for this international conference and our principal, Dr. Rakesh Sumani sir, for his constant help and guidance that has radiated as a source of energy within us. I would like to thank all the honorable national and international speakers who have blessed us with their esteemed presence and have shared their valuable knowledge with us. I feel proud and thank everyone for making this event a successful one. An event of this dimension cannot happen overnight. The wheels start rolling months in advance. We have been fortunate enough to be backed by a team of very motivated and dedicated individuals in my organizing team. So I would like to thank Mr. Amit Deshpande for making the flyer and the certificate design. I would like to thank Dr. Sonali Sonulkar for handling the registration, attendance, and the certificate aspects of the conference. I would like to thank Mr. Pradeep Chaskar for handling the logistics and IT. I would also like to thank Mr. Aditya Goge, he's from Department of Pharmaceutics, for his constant support in the IT aspects. And lastly, I would like to thank Mr. Praveen Naik for making the Zoom background and other IT related things. And finally, it's me, the coordinator, Dr. Harish Kundaikar, Associate Professor and HOD of Pharmaceutical Chemistry Department. And my role was in shortlisting the speakers, communication with the speakers, and the overall coordination of this event. So no conference is ever successful without the participation from the delegates. And I want to thank all the delegates for their registration and for their active participation. We have received around 1,495 registrations from delegates from India, Italy, Mexico, US, and several countries around the globe. And we would like to thank all the delegates who have been actively listening to all the scientific talks. Just a point to the delegates, you all would receive the participation certificates in the coming week. And I would also request all the delegates to kindly make it convenient to fill in the feedback form that would be shared in the WhatsApp and Telegram groups in a short while. Thank you all. And our principal, our convener, Professor Rakesh Sumani sir is here. So I'd request you, sir, to speak a few words.
Yeah, thank you, Dr. Harish. It was really and indeed a great pleasure for all of us to host our first international conference on the virtual platform. On behalf of entire DY Patil University School of Pharmacy and entire group of DY Patil, I acknowledge all of the speakers as well as the attendees here for giving your valuable time. Madam Mariam Degani, thank you so much for joining us on the validatory program as well. A lot of inputs we got from your talk also. Thank you so much. And Dr. Harish deserves a big round of applause for his great efforts for organizing this event. Madam, namaskar, and thank you so much for joining this event. Look forward for you to be here in our campus physically, at the earliest. Dr. Harish, please invite Madam sometimes in our campus so that we get benefit of our knowledge for our students as well. Thank you so much, Madam. And I think before we conclude for the day, hope you have shared the feedback form and other things, that formalities you must have done. Yes. Thank you so much. Yeah.
Thank you so much, sir. Thank you so much, ma'am. Thank you to all the delegates, and all of you have a very nice day. Take care. Bye-bye. Yes, hope to see you all soon for our next venture. Thank you so much. Have a nice time. Bye-bye.